Okay, hello everyone. Welcome to this first international symposium on continuous real-time biomolecular sensing. My name is Menno Prins. I'm a professor here at Eindhoven University, and I welcome all students, all researchers, technology developers, and everybody interested in the applications of continuous sensing. Together we are a big crowd, several hundred people here, people in the lecture hall, and also 300 or more people online participating in this conference. So we are a large crowd here together. In our daily lives, we all use continuous sensors. And those are primarily physical sensors. Sensors to measure continuously light, sound, motion, temperature. But in the chemical space, continuous sensing is rare. There are commercial sensors available for the continuous monitoring of glucose and pH, but that's about it. Now, fortunately, we are in exciting scientific times because we are about to make a dream come true, which is the dream to have available sensors that do not only measure glucose and pH, but are also able to measure other substances at low concentrations, like hormones, like pharmaceutical drugs, like inflammation markers, like environmental pollutants. And that's what this symposium focuses on. Sensing technologies to continuously monitor substances at low concentrations, and that means in the range of micromolar and below. And at the center of attention are the molecular and the detection principles that may serve that purpose. And the speakers will discuss sensing technologies that are able to monitor increases as well as decreases of concentration, and that do not consume any reagents, so as to be suited for long-term use. In the symposium, we'll see very recent advances and early ideas into potential applications. And we're going to look at questions like, which molecular and detection concepts are being investigated? What will be the sensitivity and the response time of the sensors? And how long could they potentially operate continuously? Which opportunities do we see and which pitfalls, and what are the next steps? The symposium idea was born in Consense and in Sensas. Now, Consense is a European research network with 15 PhD students focusing on innovations in continuous sensing. And their works over the past years will be shown at the posters and in oral presentations. Now, Sensas is the annual international student competition in the field of sensors for health, and which focuses this year for the first time on continuous biosensing. And today the 18 student teams have been testing the biosensors, and the biosensor prototypes will be on display in the poster session. The aim of the symposium is to enable as many people as possible to learn from each other in a cost-effective manner with low CO2 emissions. And that's why this is a hybrid symposium. And with a timing that allows participation from a broad range of time zones. Of course, there are still some time zones where it's now nighttime. And that's why the recordings will become available for online viewing. And it's all free of charge for everyone because it's enabled by financial support from the European Union. If this symposium is appreciated by you, then we will consider to turn it into a recurring symposium. So by email you will receive a feedback form. Please give your honest feedback, your suggestions and your opinions. And now I would like to introduce the 4 sessions of the symposium. We'll start now with session 1 on electrochemical sensing, which will be chaired by Peter Zijlstra, professor in applied physics from Eindhoven University of Technology. Then we will have food, we'll have posters and a sensor exhibition here behind the lecture hall. And then we'll have session 2, focusing on particle and fluorescence-based sensing and medical perspectives, which will be chaired by Maarten Merkx, professor in protein engineering from Eindhoven University of Technology. Then tomorrow we'll start at the same time as we are doing now, and we'll start with session 3 on plasmonic sensing and surface-enhanced Raman sensing, chaired by Sven Ingebrand, professor in microanalysystems from RWTH Aachen in Germany. Then we'll have food and posters, and finally session 4 on FRET-based continuous sensors, chaired by Michael Daniel, professor in Biomicrosystems from North Carolina State University in the United States. I asked the session chairs and the presenters to stick to the timing so that we have enough time for discussions and questions and answers. Questions can be asked here in the lecture hall, and online people can post their questions in the chat of the live stream channel. Now, before we start the first session of the symposium, I want to thank everyone who has helped to realize this symposium. So intense appreciation and thanks to the Consens team, the Sensors team, the speakers, the session chairs, the support from Eindhoven University of Technology, people online, people backstage, people on stage, people backstage, sorry, on stage, and of course the people running the audio and the video. Thanks a lot, Mark and colleagues. And let's give them a big hand. Okay, let's get started. I give the word now to Peter Zijlstra, who will chair the first session.
Yes, thanks a lot, Menno. So my name is Peter Zijlstra. I'm an associate professor here at Eindhoven University, where we work on single molecule sensing using plasmonic and photonic approaches. So I'll be chairing the first session, and we'll have 3 speakers. All 3 speakers are online today. It's a great honor to introduce the first speaker, who will hopefully appear on the screen. Yes. So the first speaker is Professor Kevin Plaxco. He's a professor at the University of California in Santa Barbara, and his research focuses on physics of biomolecular folding and applications of this folding. He's one of the fathers of the field of continuous electrochemical sensing, particularly using aptamers. He's published many papers about this and is also involved in the valorization of these techniques via various companies. So today he'll talk to us about his most recent results, and the presentation will be about 30 minutes. And as Menno mentioned, I would like to repeat, there's plenty of time to ask questions, so please keep your questions. And if you have questions online, you can type them in the chat, and Livio here will then pass them on to me. Yeah, so also the people online can be involved. So Kevin, if you're ready, then we are ready for you. So I think you can share your screen.
Good morning, everyone. Let's see. Good morning from California. Sorry I wasn't able to come out in person. I hear Netherlands is very nice in August. Can you see my slides?
Yeah, it's all good. It's all good.
Okay, great. As Peter mentioned, I'm involved in several companies that are commercializing the technology I'm going to describe today, so I do have a conflict of interest in that regard. Let me jump in, though. I always start my slide with what honestly is I think an appeal to emotion. There's a picture of a friend of mine who is now in her teens. I took this picture when she was 6. When she was 3, she was diagnosed as a type 1 diabetic. And as is true with about a quarter of type 1 diabetics in the US, she now wears that continuous glucose monitor on her arm, which reports to her smartphone about every 30 seconds what blood glucose levels are. This ability to measure glucose continuously in real time has absolutely revolutionized the treatment of diabetes. But as Menno implied, glucose is not the only molecule that one might want to measure continuously in real time. There are other metabolites that are clinically important. There are many drugs for which dosing decisions are difficult to make without accurate information on in vivo concentrations, hormones indicative of endocrinological disorders, protein biomarkers indicative of health and disease. Lots of other molecules we'd like to measure continuously in real time. The problem historically, however, has been that the glucose sensor, the one truly successful example of a continuous real-time in vivo molecular monitoring system, it relies on the chemical transformation of glucose by an enzyme. That is, it's critically dependent on the chemical reactivity of its target, and that renders this approach to real-time in vivo molecular monitoring fairly difficult to apply to other targets. And indeed, in the almost 50 years now since the first in vivo glucose sensor was described, this idea of enzyme-based electrochemical sensors has only been expanded to of order a half dozen successful examples in which measurements were performed in vivo. That highlights what the goal of my research group is. We're trying to develop a platform technology that can, like the glucose monitor, measure specific small molecules with high frequency in situ in the body, but that a technology that's agnostic to the chemical reactivity of the targets, and therefore, in theory at least, should be generalizable to many targets. And what makes us think that's a solvable problem is that it has, of course, actually been solved already. It's just that it's been solved by evolution. You're all full of receptors that are responding to specific molecular cues right now just by binding those targets, not by chemically transforming them. To my looking at the problem, evolution had to invent two tricks to solve this problem that I'm going to illustrate here using a cartoon of a gated ion channel. Of course, the first trick that evolution uses to perform molecular monitoring in vivo is the combinatorial chemistry of biomolecules. The combinatorial chemistry of proteins and even of nucleic acids is so great that one can make receptors that will bind almost any arbitrary water-soluble target. And of course, that idea has been around for a long time, the generalizability of biomolecular recognition is really what's prompted 50 years of research on biosensors. But recognition alone is insufficient. Evolution had to invent a second trick to perform in vivo molecular measurements, and that is typically using binding-induced conformational changes to transduce binding events to outputs. So in the gated ion channel, for example, when a neurotransmitter binds the channel, that changes the conformation of the channel, in this specific case, opening up a pore that allows an ion current to flow. More generally, though, if you look across all chemoperception systems in biology, you see that receptor binding is coupled to a big change in receptor physics, typically a conformational change that's used to produce an output in a manner that, again, is independent of the chemical reactivity of the target. My background is molecular biophysics, and so when I entered into the biosensor field, tackling the signal transduction problem was our entree point into the field. So how do we do that? Well, first, let me take a moment to mention how, in my group, we solved the first trick there, the combinatorial chemistry trick. All of the work I'm going to share with you today employs DNA aptamers as the receptors. So that is synthetic DNA molecules generated by an artificial in vitro selection system to evolved in vitro to fold up like a protein does and bind a specific target. So that's the combinatorial chemistry side. How do we solve the signal transduction problem? Well, the folding of aptamers is generally fairly cooperative. It's cooperative enough that if we destabilize the native state via mutations anywhere on the chain, and typically for convenience we'll make those mutations as truncations to one or the other end of the chain. If we destabilize the chain enough to push the folding free energy positive, the aptamers tend to globally unfold. Now, if we do this delicately, if we destabilize them enough that the folding free energy is unfavorable by about 6 kilojoules per mole, then under physiological conditions, the chain spends, any given molecule, any given chain, will spend 90% of its time unfolded, expanded, highly dynamic, but it still spends 10% of its time in the native fold. And for aptamers, the time scale with which these two states interconvert is typically milliseconds. So again, while any given chain spends 90% of its time unfolded, dozens of times per second, it's still sampling its native state. How does that help us here? Well, of these two ensembles, of these two states, the unfolded ensemble and the native state, it's only the native state that has the right arrangement of functional groups to bind the target. So if I throw the target into this equilibrating mix, it will preferentially bind the right-hand side of the equilibrium and therefore drag the equilibrium back over. These destabilizing mutations that unfold the chain also have the property of introducing into the system a coupling of recognition with folding itself. It was that recognition, that the realization that these stabilizing mutations are now coupling molecular recognition to the biggest conformational change there is that got us moving into the biosensor field, as we recognize that coupling recognition is such a big change in physics, should be a great way to produce really robust outputs that we could measure. Now, one way we could monitor folding and therefore monitor the presence of the target would be to put fluorophores and quenchers, for example, on the chain, and thus causing the binding-induced folding event to produce a change in fluorescence output. And honestly, in our lab, we do that with some frequency because fluorescence experiments in the lab are really quite straightforward. In the real world, fluorescence becomes more difficult. Performing fluorescence measurements in vivo is complicated by the fact that many things in vivo absorb photons, scatter photons, and many things in vivo fluoresce themselves. And so instead, for our in vivo sensors, we developed an electrochemical method of monitoring folding that, again, then reports on the presence of the target. To do that, we attach one end of the aptamer to a gold electrode using alkane thiol gold sulfosimilar monolayer chemistry. On the other end of the aptamer, we place a redox reporter, typically methylene blue, but something that gives up or takes on electrons easily. In the absence of target, the aptamer is unfolded. The methylene blue typically approaches the surface only rarely, and the electron transfer rate is slow. Binding-induced folding will typically push the methylene blue down to the surface and accelerate the electron transfer rate. Alternatively, it could pull the methylene blue away from the surface and decelerate electron transfer rate. Either way, the binding-induced conformational change changes the electron transfer rate relative to that seen in the unfolded state. And then we use any of a variety of electrochemical techniques that are particularly sensitive to electron transfer rate. I believe all of the data I'll share with you today were performed using square wave voltammetry. So an example of that, we put an unfolded aptamer against the antibiotic kanamycin on our electrodes, sweep the potential, measure the resulting current. As we add more kanamycin, the signal increases. These are quantitative, obviously, and reasonably accurate and reasonably precise. Here are data collected in my lab, fairly typical data. These are hand-built devices built by my students. Honestly, we don't have very strong quality control efforts. These are pretty casually built, and yet our sensor-to-sensor reproducibility is pretty decent. So this is 4 independently fabricated sensors. Through the clinical range in gray of vancomycin, we're seeing accuracy and precision of about 10%. Okay, back to my story. Remember, my goal is continuous, or at least high-frequency, real-time measurements of rising and falling concentrations. And if you look at the architecture, you can see that this sensor approach should support at least continuous and real-time measurements. For example, the association and dissociation rates, timescales for small molecules binding aptamers at clinically relevant concentrations is typically faster than a second. Likewise, the folding and unfolding rate of the aptamer, that time scale is typically milliseconds. And so, in fact, these sensors respond to rising and falling concentrations. They equilibrate to rising and falling concentrations, at least of small molecules, typically with speeds faster than the few seconds it takes us to perform the square wave voltammetry scan. Now, I also want to perform those continuous real-time measurements in situ in the body. For that, the sensor needs to be selective enough to work in complex biological fluids. We thought when we started down this road that it should be, because again, the signal transduction mechanism we're using is the same one that biology's invented to perform signal transduction in you. Consistent with that, this was some old work that my group did in collaboration with Tom So, who's also speaking in this series. Here we placed a sensor against the drug of abuse cocaine in a microfluidic chamber, and in red, through which we flowed a sample of undiluted blood serum, and in red, the concentration of cocaine in that blood serum stream is fluctuating, and in blue, you're watching the sensor following the rising and falling cocaine concentration. I'll add that these data are a little bit unfair to our sensor. This 90-second or so time scale equilibration time for the rise and the fall is actually the flush time of the chamber. The sensor itself responds in faster than the few seconds it takes to perform the square wave voltammetric scan. Okay, so here we've got a sensor that works continuously and in real time in undiluted blood serum. We were pretty excited about that at the time, but after thinking about it a little while, we realized that this is actually a fairly artificial system. A blood serum is what you get if you take a test tube full of blood and you let the blood clot, or you remove the solid clot, throw it away. The serum is the straw-colored fluid left over. So when you think about it, you never have a flowing stream of blood serum. That's completely artificial sample environment. Flowing stream of whole blood, yes, that's where I want to be. Not in blood serum, but I want to be in a flowing stream of whole blood in you, in vivo. So after getting these results, we then transitioned to performing experiments in animals. These sensors, by the way, can be made quite small. We'll typically fabricate them on 75 micron diameter gold wires, a few millimeters long. 75 microns is a little bit thicker than my hair, a little bit thinner than typical black hair. So they're pretty small. Conversely, we're putting these sensors, we're doing our in vivo studies largely in rats. Rats are also pretty small. And so in vivo, intravenous sensor placements in rats mean placement in the jugular. And the next picture is a picture of that. So here's an anesthetized rat. We've exposed its right jugular and then slipped a sensor into the right jugular via a catheter. And then in the left jugular, we've placed a second catheter that allows us to dose the animal with the target drug. And does it work? Well, sort of. So here's a sensor for the chemotherapeutic doxorubicin, which we placed again in the jugular of a live rat. Here we've dosed the rat with 4 doses of doxorubicin, and you see the sensor responding. We dose the rat, the concentration rises, and then as the rat metabolizes and excretes the drugs, the concentration falls. The problem is there's no drug here. This is just the sensor drifting. So whereas the sensor is really quite stable in blood serum at room temperature, in whole blood, in vivo, at body temperature, it drifts significantly. Now, when faced with drift, a common engineering approach is to place a reference sensor adjacent to your actual sensor. That is a reference sensor, a sensor that responds to the environmental effects but doesn't respond to the thing that you're measuring, and therefore provides a means of measuring what the drift is and correcting it. We can't do that because this drift is quite variable from one sensor placement to the next. So if we put 2 sensors in the same animal, they don't drift in concert. Instead, to solve this problem, Scott Ferguson, who at the time was a graduate student working again with Tom So and collaborating with my group, Scott came up with a clever method of using the sensor as its own reference. To see how to do that, we'll look back at the architecture here. Again, target binding is signaled via a change in electron transfer rate. Square wave voltammetry is so sensitive to transfer rate that we can make it more sensitive by tuning the square wave frequency. We can make square wave voltammetry more sensitive to rapid electron transfer or more sensitive to slower electron transfer. If it's more sensitive to rapid transfer, then target binding causes the current to increase because the bound state transfers more rapidly. Conversely, though, if we tune it to be more sensitive to slower electron transfer, target binding causes the signal to decrease because in that circumstance, the signal's more sensitive to the unbound state. Here's an example of that, again, with our kanamycin sensor. At a square wave frequency of 500 hertz, the signal increases as we add target. At a square wave frequency of 60 hertz, the signal decreases as a function of target. How does that help us? Well, back to that in vivo doxorubicin data. At 120 hertz, it's signal on. At minus 10 hertz, it's signal off. But the 2 signals drift in concert. So we can use the 10 hertz signal as a reference to remove the drift from the 120 hertz signal. And when we do that, here it is. We have what I want. This is continuous, few seconds resolved at least, high frequency, real-time measurements of this drug in situ, in the jugular vein of a live animal, using a technology that does not rely on the chemical reactivity of the target. It relies merely on the availability of an affinity reagent against the target. And therefore, the approach should be general. And it does seem to be pretty general, because since we reported this first sensor now 7 years ago, we've expanded the platform quite a bit. Here's in vivo data measurements of another chemotherapeutic. Here's another chemotherapeutic. These are all in the rat jugular. Here's an antibiotic. Here's another antibiotic where we did ex vivo blood draws. Oh, quick note, again, the title of my talk is Recent Advances. The yellow star means something we published in the last year. Red stars are going to denote data that are unpublished. I wanted to emphasize how quickly this field is starting to move. Here's another antibiotic. Here's another antibiotic. Here's another antibiotic. Here's an antifungal. Here's the drug of abuse cocaine. Here's the metabolite tryptophan, again with some ex vivo validation measurements. Another metabolite, another metabolite, another metabolite, another metabolite, another metabolite. That's it. We're up to, as of last week, we're up to 18 different sensors that we've placed in the jugular veins of rats, in every case performing typically few seconds resolved measurements. We can push the measurements, by the way, down to sub-second using chronoamperometry, although sub-second measurements honestly are overkill resolution for most of these targets. So a fairly general platform. Now, so what could we do with this? I mean, we've got what I wanted, a platform technology that in theory can measure any molecule in the body with high frequency and in real time, and in practice can measure lots of molecules in the body. What can we do with this? Well, obviously there are clinical applications. And as I mentioned, I work with some companies that are commercializing this. Predominantly Nutromics, a company in Melbourne, Australia, has about 50 full-time people working on the clinical applications of this technology. In no small part because now companies are pursuing the development of this technology, though. My own group has shifted away from technology development and moved towards the scientific applications of this new tool. As an example of what we can do scientifically with this tool, first, I'm presenting here a pharmacokinetic cartoon of the sort you'd see in any pharmacology textbook, a cartoon of drug absorption into the body and drug elimination. Now, it's almost always a cartoon in the textbooks because historically we've measured drug concentrations in the body via blood draws and benchtop analysis, approaches that are so cumbersome that the data are generally very sparse. This is an example from our lab where we did 7 blood draws on a rat after an intramuscular injection of tobramycin. We're only allowed, by the way, to make 7 blood draws before the animal's blood pressure drops too low, and animal welfare concerns preclude any more measurements. With 7 data points, we can see the adsorption phase here and the elimination phase, but we don't constrain the kinetics of either of those phases with very much precision. In contrast, when we're making a measurement every 6 seconds or so, we constrain those time scales to a precision of within about 2%. So this is really just unprecedentedly high precision pharmacokinetic measurements. Indeed, actually, our data density and our data quality here are great enough that we should actually constrain these timescales to precision of better than 2%. The issue is, at this level of precision, the models we're employing, the standard pharmacokinetic models, start to fail. Standard pharmacokinetic models assume, for example, that elimination is a first-order process with a first-order rate constant. But the rate constant isn't really constant. And for this drug, elimination is via the kidneys, and kidney function varies. So recently, we've been using our data to explore these ideas. In one sense, kind of obvious ideas that physiology changes often over hours time scale. So it had always been assumed that drug elimination rates would change, but historically no one's ever had the ability to measure how fast elimination time scale changes. Here's an example we published last year, earlier this year, on tobramycin, where we're looking at the proportionality between concentration and elimination rate and how that proportionality changes over the course of a few hours. And for most of the animals we studied, it's changing by about a factor of 2 or 3 over the course of a couple of hours. That's because this drug, again, is eliminated via the kidneys. These animals are under anesthesia, no IV drip, their blood pressure is dropping, their kidney function is falling, and so the drug's staying in their bodies for longer than it would otherwise. This is tobramycin. Here's some unpublished data from vancomycin. Like tobramycin, it's eliminated via the kidneys. It's got a similar half-life to tobramycin, and therefore, not surprisingly, we're seeing the same sorts of few scale changes, 2 to 3-fold changes in elimination proportionality over the course of a few hours. We're making these measurements in real time. So another thing we can do with this technology is use it to close the loop and perform feedback controlled drug delivery, whereas we take real-time measurements, pump them into a controller, typically just a simple PID controller, use that controller to then control a syringe pump that's delivering the drug to achieve whatever desired concentration we want. Here's an early example, feedback control over plasma tobramycin levels. So maintaining pretty much perfectly constant drug levels in vivo, couldn't be done before. Here's another example, feedback control over plasma vancomycin. So far everything I've shared with you were measurements performed in the rat jugular, but these sensors are quite small and can be inserted pretty much anywhere in the body. The next compartment we started to explore was intracranial measurements. Specifically, here are measurements of the anesthetic procaine in the lateral ventricle of a rat. These are 2 measurements performed a week apart in the same animal. This drug is eliminated enzymatically, and therefore its pharmacokinetics don't vary very much, even over the course of a week. Here's a control experiment. The drug doxorubicin is known to not cross the blood-brain barrier, and indeed, when we put a very sensitive sensor to doxorubicin in the brain, we don't see any doxorubicin crossing at all. So sensor insertion is not disrupting the blood-brain barrier. Here's a still unpublished results where we placed sensors in both either the lateral ventricle or the hippocampus of a rat and showed for the first time, again, not rocket science. It was expected that different parts of the brain would receive drugs with different rates. Historically, no one had ever really been able to measure that before, and here we are measuring it. We're seeing small, so these bars reflect 95% confidence intervals, small but statistically significant differences in the time it takes to reach the maximum concentration, in the time it takes to pass through a 10 micromolar threshold. So again, an expected result on one hand, the brain is heterogeneous, different regions should have different pharmacokinetics, but no one had really ever been able to measure them before. With these in-brain measurements, we can perform, because the skull is so solid, the sensors are placed very solidly in the brain, so we can perform these measurements on, well, initially awake animals. Procaine's an anesthetic though. So in red here, we have how much the animal's running around the cage, and as the in-brain procaine concentrations rise, the animal starts to fall asleep, and then as metabolism eliminates the drug, the animals wake up again. I like this slide. There's probably 20,000 measurements here. It would have been very hard to do this with HPLC, with standard benchtop approaches. Here's the correlation, by the way, between how much the animal's running around the cage and the concentration of procaine in the brain. And what we find is across these 9 animals, the correlation is statistically significant for 8 of them. Now, these are not the strongest correlations in the world, but this is really unprecedented in the field of psychobiology. Historically, the folks in that field have performed their studies by giving the animal dose X and seeing behavioral response Y. And there's so many steps between dose and response that correlations are generally quite weak. Here we're quantitatively measuring the concentration of the drug near the site of action of the drug and correlating that with behavior. And those two are so much better linked that, again, we see statistically significant correlations for most individual animals. So that was procaine. More recently, we've developed a very sensitive sensor to cocaine, put that in the lateral ventricle of an animal, And you see, in contrast to procaine, which is an anesthetic, cocaine is a stimulant, so the animal runs around a lot. This is really one of our better sensors. At a 3 sigma confidence level, the baseline noise here is 15 nanomolar. It's really an excellent sensor. Our aptamer collaborator, Zhao at North Carolina State, who coincidentally invented EAB sensors back when she was a postdoc with me a long time ago, she's making really amazing aptamers now. We're, again, collecting these in-brain measurements in real time, so we can also perform feedback control over concentrations in the brain, not just in the vein. I think this is going to be a powerful tool. Right? Historically, there really wasn't any way of controlling the concentrations of drugs in the tissues other than by varying dose. And the linkage between dose and concentration is so variable. That's really terrible control. Now we can actually actively control the concentrations of drugs in various parts of the body with really quite high precision. I think that's going to be an important tool to help us understand and elucidate pharmacokinetics, pharmacodynamics. All right, so far all the measurements I shared with you were performed using a single sensor in a single compartment. But of course, we can put multiple sensors in a single animal and measure, therefore, with high precision how rapidly drugs and metabolites are moving from one compartment of the body to another. Here is our first example. Actually, this is impressed now. It should be a yellow star. Simultaneous measurements of the antibiotic vancomycin in the brain of a rat versus in the vein. It does not, it crosses the blood-brain barrier, but doesn't cross it very well, so you see the in-brain concentrations stay quite low. That's one example. More recently, we've really started to focus on simultaneous measurements in the plasma and in the subcutaneous interstitial fluid. 2 reasons for that. One, the interstitial fluid is the site of action in most drugs. Most drugs, their site of action and their site of toxicity is in the solid tissues, not in the blood. So understanding pharmacokinetics in the interstitial fluid seems to us to be of great clinical import. Additionally, we'd like this technology to really have significant clinical impact. And it will have much greater impact if we can perform the measurements using a small needle placed in the subcutaneous space, analogously to the glucose sensor, rather than sensors placed in the vein. That said, even though drug measurements in the ISF are probably clinically more important than plasma drug measurements, everything clinicians know is based on plasma measurements. So while I think ISF measurements will be more valuable ultimately than plasma measurements, the clinicians want to know what plasma drug concentrations are. So to pursue this wearable device that's making ISF measurements, we really need to understand the relationships between drug concentrations in the ISF and drug concentrations in the plasma. So we've just started down this road. Here's one example. I think we've got 4 or 5 different drugs we've done this for so far, none of which are published. So this is simultaneous measurements of the chemotherapeutic methotrexate in the plasma and in the subcutaneous ISF. Here's another example, another chemotherapeutic, doxorubicin. In this experiment, we perform feedback control over the plasma drug levels to keep those constant. And then we just passively place 2 sensors in the subcutaneous space to watch as the drug concentration in the ISF equilibrated with the drug concentration in the plasma to understand how quickly, if we're performing feedback control to maintain constant levels in the plasma, how quickly do the tissues achieve the same level? That was one question. The answer is relatively quickly. Another question was, how reproducible are our in vivo sensors? These 2 sensors are placed in different spots in the sub-Q space. They might have very slightly different kinetics. You see they have very slightly different kinetics, but they should equilibrate to the same point. And to within our typical error of about 10%, they do. That was doxorubicin. Here's paired measurements of vancomycin in the sub-Q space. Again, slight differences in the pharmacokinetics in the 2 positions, but really quite slight. The 2 sensors generally follow one another quite well. We've recently started modeling this intercompartmental transport from the blood into the solid tissues. To do that, the first thing we did was fit the blood data, the plasma data, here to a 2-compartment model, and then assume that this red curve is reflecting the true plasma concentration. And then we fit the simultaneous subcutaneous measurements First, we fit it to just an extremely simple model, a model that assumes that only the plasma and the ISF matter, that we only have these two compartments, and that it's simple passive diffusion between them. So there's only a single fitted parameter in this model. Considering there's only a single fitted parameter, it fits the data reasonably well. If we had, as is typical in this field, 5 data points, you'd probably say it fit quite well. Clearly, though, it's got a systematic offset. So we next looked at a more sophisticated model in which, due to physiological effects, the rate of transport is asymmetric in and out. And you see in purple, it's hard to see the curve. It fits the data. This blows my mind how well this simple model fits the data. Again, this model only has 2 fitted parameters in it, and yet it's fitting this rather complicated set of curves really quite well. Still, this result surprises me. So that was vancomycin in the sub-Q. Here's doxorubicin. We've looked at a number of different doses. The relationship between drug exposure, the area under those concentration time curves versus dose in the plasma is terrible. That's why we're doing this. The relationship between dose and drug exposure is very poor, so we want to actually measure exposure so we can adjust dose. Not surprisingly, the relationship between dose and exposure in the ISF is also very poor. But this result delights me. Exposure in the ISF, which is quite easy to measure with minimally invasive sensors, is highly correlated with exposure in the blood that clinicians right now care about. So suggesting those wearable subcutaneous devices will be giving us clinically important information. 2 last things. These subcutaneous measurements are much less invasive than intravenous placements. That's allowed us to do a couple of things. First, it's allowed us to do long duration experiments on awake animals. So here's an animal with a subcutaneous sensor in it. It's a little sleepy because they're nocturnal and it's daytime. Here's some of the data from those experiments. So this is a vancomycin sensor left in the animal for 24 hours. We challenged the animal twice with doses of the drug, and you see after 24 hours it's still responding well and giving us good return to baseline. So as I mentioned, the sub-Q sensors being much less invasive allowed us to do 2 things. First, long duration measurements. The second thing is they allowed us to finally move to animals other than rats. And specifically with these subcutaneous measurements, sensors, we've made our first in-human measurements. So this is in a human arm, subcutaneously placed sensor following a large oral dose of the amino acid phenylalanine. And so we're seeing the drug being absorbed from the gut and into the bloodstream and then metabolized. And lastly, yes, I did put my money where my mouth is. Oops. Pro tip, it turns out phenylalanine dissolved in beer tastes absolutely terrible. And my wife took this picture because she thought that my expression was so amusing. All right, that's the last bit of science. Let me close with the acknowledgement that I, of course, haven't done any of the experiments. I've been blessed with having a really strong research team now for many years. Let's see, and I mentioned we started down this road a long time ago with Tom So, who was our collaborator at UCSB before he moved up to Stanford. And I don't know if Tom's joined us yet this morning, but he is joining us soon. And with that, let me close and happily take questions.
All right, thanks. Thanks a lot, Kevin. Does it work? Hello.
Hello? Yes.
Now it works.
I can hear you.
Yes. Bit of echo. Thanks a lot for this inspiring start of the symposium. We have time for questions. Who would like to start? Yeah, I will bring the microphone.
Thanks a lot, Kevin, for your very interesting presentation. You started off with the challenge of drift and then the solution of having the square wave voltammetry. And so apparently there's a necessity to correct for drift. But what is really the origin of the drift? Because you're also showing data 24 hours vancomycin, and there's really no change of noise, it seems. And so the signal, it looks like as if the sensor is completely unaffected by 24 hours of exposure. So is it really necessary to correct for drift? And if it's necessary, what is really happening? What is really causing this drift? When we use square wave, it really is necessary. I didn't show you the raw peak. Well, I
In vivo data, remember, for doxorubicin.
Kevin, your the sound dropped. Can you repeat your answer?
So I showed you that first in vivo data set with doxorubicin. I showed you the peak currents as a function of time in the rat, and they drop dramatically. Over the course of those 24-hour experiments, we lose about 80% of our current. This drift correction mechanism is correcting for that and still giving us good, accurate return to baseline, for example. So it really is critical to have that drift correction. And the subcutaneous measurements just have such good signal to noise that the noise there is mostly from the animal running around the cage, which doesn't change very much. But the signal to noise does get poorer after 24 hours because we've lost 80% of our signal.
Okay, so it
Mechanistic.
Yeah.
Go on.
No. So you say it does affect the noise level, although it's maybe not visible due to other noise sources. But then the origin of it.
We've recently published a paper where showing that if we use non-natural backbones, so-called xenonucleic acids and X and A, which are very resistant to nucleases, the drift is reduced by about a factor of 5. So our strong suspicion is that it's mostly nuclease-driven degradation of the DNA aptamer, which, again, we can surmount by using non-natural backbones. There are a number of groups now that are starting to select aptamers using non-natural backbones, so I'm quite excited about that.
So maybe one day the drift correction is not needed, will not be needed anymore.
That's right.
Okay, thanks.
There are other ways, there are other ways of doing the drift correction. We can also use, say, electrochemical impedance spectroscopy to pull out the transfer rate itself. And transfer rates are independent of how many aptamers you've got because it's measured in seconds. So even if you lose aptamers, it doesn't change the transfer rate. So there are electrochemical methods that are intrinsically drift resistant. You still have problems with signal to noise, though, as you lose aptamers.
Okay, thanks a lot. I first have a very simple question, Kevin. Could you stop your screen share so we can see you full screen here? And then in the meantime, are there other questions in the audience? I see one in the back.
Thank you very much for the presentation. Very nice. I liked a lot your model. How did you fit indeed the very easy equation? But you said that initially, it's beautiful that it looks very easy. But you said that firstly you tried to fit it with this passive diffusion equation. Then it appeared that the constant for the rate should be different. But
Yes.
what would be the reason that the rates, like for income and outcome, are different?
Yeah, a very fair point. There is no real barrier between the capillaries and the tissues. The capillaries are leaky. So it's not like there's a membrane there across which active transport is occurring. So this too caused me to scratch my head. We think there's two possibilities, one of which I'm happy with and one of which I'm less happy with. Before I launch into those, I'll say that the differences in the rates is typically only sort of 10 to 25%. It's not huge. So it could just be bad sensor calibration. If the calibration is off by 10 or 20% between the plasma and the ISF sensors, you'll get that. Alternatively, though, it could be physiology. The drug there, vancomycin, has a ionizable group with a pKa right around neutral. And because of metabolism, the tissues are in dynamic equilibrium. They are somewhat more acidic than the blood is. So that could be a physiological drive to drive more excess concentration of the drug into the ISF. So there is a potential physiological explanation, but I readily admit, given that the difference is only like 10 to 25%, it could be bad calibration.
Yeah, okay, thank you very much.
All right, who would like to continue that side? Need to make my way.
Yeah, thank you for a very nice presentation. I just wanted to ask about the targets, because most of your targets are drugs or some small molecules. Do you envision, or maybe you already tried some targets, more like protein targets?
We've done one protein in vivo so far, platelet-derived growth factor in the jugular rat. We haven't published it. We're currently working on several more sensors for proteins. One problem with proteins is that the on and off rates tend to be slower, so sensor equilibration time becomes rate limiting. But for some of the proteins we're interested in, the time scale of that's only a few minutes. So we won't have seconds resolved data, we'll have few minutes resolved data, still of significant value relative to how fast these biomarkers change. So absolutely we can do that, and we're moving down that direction. One question is how fast these markers get into the ISF. Will we have to put the sensors in the blood to measure them, or do they get into the tissues fast enough that that works? Unknown. The answer to that question is unknown. We look forward to addressing it.
Okay, thank you. If I may ask one more question, short one. I also wanted to ask about the stability and those aptamers or xenonucleic acids that you mentioned, because I can imagine that that also changes your the binding of to your aptamer when you introduce some different
Backbones.
Yeah.
Yes. Our assumption is that in most cases, one will have to select the aptamers as XNAs. You can't just, in general, you probably can't just convert a DNA aptamer into an XNA aptamer and hope that will work. And there are several groups now in the world who are selecting aptamers out of non-natural, out of about 4 or 5 different non-natural backbones. So that's a rapidly emerging technology. The one example we used was an aptamer that we were pretty sure that we could. It's an aptamer against the aminoglycoside antibiotics. It's of known structure. And by looking at its structure, we were pretty sure we could convert the original RNA aptamer into both a DNA aptamer and a 2-methoxy-RNA aptamer. And all 3 of those worked in that one special case. So we could do the direct comparisons there.
Thank you.
All right, thanks. I would also like to give the people online a chance to ask a question. Are there questions online? There should be a second microphone for Livio, or not? There is, or?
Hello. Okay. So there are a few questions online. So first one, Tony says that many xenobiotics, such as drugs, exist in largely as protein-bound fraction in blood. And he's wondering if the electrochemical aptamer-based measure free or the total fraction of those drugs.
Our presumption is that it's almost always measuring the free, because it's the free drug that will be in equilibration with binding of the aptamer. The vancomycin case may be a counterexample because vancomycin is big relative to most small molecule drugs. Its molecular weight 1,500. So we have been curious. We don't know this yet, but we have been curious as to whether or not the protein-bound drug still has the aptamer binding epitope expressed. But in general, we're pretty confident that we're just measuring the free drug.
Okay. Patrick also is congratulating for the presentation. And he is wondering if these sensors have been already commercialized, and if not, why are the main reasons why it has not happened yet?
There's a 50-person strong company in Melbourne, Australia, Nutromics, that's working on the clinical applications of this. Their first product will be a vancomycin sensor. That drug is particularly difficult to dose safely and effectively. So the clinical reception for that. Now, it can be difficult to get clinicians to adopt a fundamentally new technology. Most clinicians really don't know what they would do with real-time measurements. Vancomycin, fortunately, though, is a counterexample. Every clinician I've talked to is excited about the ability to measure that because they all know it's very hard to dose that drug safely and effectively. Technologically, I'm not too concerned about things. The company's making good progress on all technological fronts. Convincing the FDA to accept a fundamentally new technology is something I personally don't know much about, so that's something I worry about.
Okay. Fabienne is asking how good the translatability of this of sensing mouse to humans. So she's, I think she's wondering how good is this how translatable it is from mouse to humans.
You know, the folks who do drug development have to worry about that all the time because drugs are targeting some specific receptor, and the receptor in a rodent's not the same as the receptor in us. So animal models are not good proxies for humans in drug development. In contrast, here, the sensor's measuring the same target in both our rats and in me. And so that translation is really trivial.
Okay.
Last question, Livio.
Okay, just one more question then. And Santino is asking if the drift could also be due to the thioreduction?
Yeah, so we attach our aptamers to the electrode surfaces using thiol gold chemistry. If we keep the potential window that we scan, if we keep that fairly narrow, we don't see much loss of monolayer. If it gets a little too broad, then we electrochemically desorb the alkane thiol. So that has been a problem in the past until we learned to keep our potential windows narrow.
Okay, so who would like to have the last question in the room? I see in the corner, Rachel.
I guess it's okay. Thanks. My question is about all the different complex matrices you worked in. You talked about blood, plasma, subcutaneous fluid. Have you explored other ones? And what are the next ones you think you'll try to tackle next?
So in vivo, we've done plasma, so blood. We've done interstitial fluid in both the subcutaneous space and intermuscularly. We've done cerebral spinal fluid in both the lateral ventricles and the solid brain tissues. All of those worked well. Some years ago, there was a company that has since failed, sadly, that was trying to adopt this technology to measurements in sweat. And when they first reached out to me and asked if I thought the sensors would work in sweat, my naive response was, of course, because, I mean, for God's sakes, they work in blood in vivo. Sweat seems far less complex than that. That was naive. In sweat, the pH and the ionic strength and the ionic composition all vary really dramatically. And our sensor is sensitive to all of those things. For example, the methylene blue reporter transfers 2 electrons and a proton, so its kinetics are pH sensitive. So that made sweat a very difficult matrix. In contrast, in the blood, in the CSF, in the ISF, pH, ionic strength, and ionic composition are all really tightly controlled. So normal physiological fluctuations in any of those don't significantly degrade sensor performance in vivo. But ex vivo in sweat, probably in saliva, things get much more complicated.
Perfect. Thanks.
All right, so please join me in thanking Kevin again for a great presentation.
Thank you, everyone. It's my pleasure. So again, sorry I couldn't join you in person.
Thanks a lot for being here. And then we move on to the next talk. And the next talk will be by Hossein Zargartalebi. I saw you online, so you are there. Welcome. So Hossein is a postdoctoral fellow at the Northwestern University in a group of Shana Kelly. He got his PhD in mechanical engineering from the University of Calgary in 2022, and then he went on to do a postdoc in pharmaceutical sciences at the University of Toronto. And his research is about electrochemical sensing of proteins and small molecules. And I assume that is also what you will speak to us about today, Hossein. So please go ahead. The floor is yours. You can share your screen, and then hopefully everything is good and ready to go.
All right. So can you see my screen?
It's coming up. It's still black, but let's see what's happening. Not yet.
So I will stop sharing and share again, maybe.
Yeah. That did not help much because it's still black. Any advice from the support here? No, no advice. Oh.
All right, let me try again.
Are you sharing your desktop, Hossein, or are you sharing your PowerPoint?
I'm trying to share my PowerPoint, but it seems like...
Yeah, if you share a desktop, maybe that will work.
Yeah, so...
The only drawback is that you will not see us, but...
So just a moment, sorry for that.
That's okay. I find it difficult to advise because I cannot see what is happening.
Yeah.
Or is it an idea that we swap the two talks so you can take a look at it? Would that help, or?
So I try to share my screen in another way, but I don't know what's happening here. Maybe the file is a bit large.
If you share a desktop, that should not matter.
So how can I share my desktop? So
Yeah. Well, it's the same button share, and then you'll have like a few big squares that show.
so it shows. Okay,
Yes,
so
there it
what
is.
about now? Can you see my screen?
Yeah, we can. If you make it
Perfect.
full screen, then we're ready.
So can you hear me okay as well?
Yeah, your sound is perfect.
Perfect. All right.
Yeah, all good.
Oh, perfect. Yeah. Okay, so thank you so much for having me here today. I would like to thank you all, the organizers of this symposium, especially Professor Prins, for inviting me for this presentation. This symposium regarding the continuous monitoring, which I think is very important topic in the biosensors. I'm really excited to share with you my recent work that I've done at Kelly Group at the Northwestern University, focusing on real-time monitoring of the proteins in vivo using active reset methodology. So I would start with the rationale behind this project, which is why continuous monitoring. A simple answer to that is kind of empowering the health system with the continuous monitoring gives us the opportunity to diagnose the onset of diseases with the early detection of diseases, kind of helps us in improving the personalized treatment, understanding disease progression, and also enhancing the patient outcomes. So altogether, as Kevin just mentioned, that continuous monitoring revolutionized the research in healthcare and will save millions of the lives. So currently there are a lot of technologies available for in vivo real-time monitoring. Some of them have been commercialized, tracking vital signs such as body temperature, heart rate, electrocardiography, muscle action, respiratory rate, and blood pressure. There are also some beautiful works tracking small molecules in a real-time fashion, such as drugs, neurotransmitters, and metabolites, electrolytes, and so on. Here are some examples of the wearable implantable sensors and devices that have been developed for measuring metabolites such as glucose lactate, electrolytes such as sodium and potassium, neurotransmitters, hormones, blood pressure, body temperature, and so on. So all of these devices and technologies have been developed for tracking either vital signs or small molecules. The question is how we can perform real-time monitoring of proteins in vivo. To answer these questions, I need to clarify more the challenge here. For the small molecules, such as drugs, neurotransmitters, and metabolites, the affinity of the receptors ranging between low to the medium values, so that we have fast dissociation rate or moderate the dissociation rates, meaning that when the analyte and receptor attach to each other, they can easily dissociate from each other, so that we can have the real-time monitoring of such kind of molecules. But for the proteins, the story is a bit different. So for the proteins, we need to use high affinity receptors with slow dissociation rates. We know that the physiological level of proteins in the body is very low, so that we need to use high affinity receptors, which gives us the opportunity to have sensors with high sensitivity, false negative, and high specificity, so that false positive, to be able to track such kind of protein. So for the high affinity receptors, let's say with a KD of 1 nanomolar, we will have a dissociation rate approximately in the range of 10 to the power of -5, which gives us the complexation half-lives of 20 hours. So it means that when the analyte and receptor attach to each other, so it takes a long time to dissociate these two from each other. That makes kind of real-time monitoring of proteins impossible. So for solving this problem, we came up with an idea using such kind of high affinity receptors to track proteins in real time. All right, before jumping into the how these kind of sensors are functioning, I need to rewind the methodology that we developed in our lab, and which was kind of the motivation that we use for continuous monitoring for the proteins, which we call it reagent-less molecular pendulum sensing. Regarding the reagent-less sensing, Kevin Plaxco group and Thompson group have done beautiful works. So here we have done reagent-less molecular pendulum sensing for targeting proteins. Generally, in such kind of methodology, we have double-stranded DNA attached to the electrode surface. The ferrocene redox reporter will be used for transferring electron, which is attached to the distal end of the DNA. And we can use several types of receptors like antibody, aptamer, and so on. So in such kind of, let me turn on my laser pointer here. Okay. Because the sensor has negative potential, when we apply a positive potential to the surface of the electrode, we are bringing the whole sensor to the surface, so they're going to be an electron transfer between the ferrocene and the electrode. So the kinetic of the electron transfer is going to be different between the unbound and bound case. As you see here, the current decay is different when we are in the unbound state and when we are in the bound state. So based on this difference, we can find out the concentration of the protein. So we continued this work and came up with an idea of applying an alternating potential through which we can oscillate the sensor. With the oscillation of the sensor, we are applying a high energy to the receptor and the protein and kind of inducing the dissociation of the protein from the receptor, that we call it active reset. So by performing active reset again and again and again, we can perform continuous monitoring of the protein. We can track the level of proteins, both increase and decrease, as you see here. All right, as a starting point, we compared the active reset versus passive reset by targeting the MPO protein. So for the passive reset, so we have done this for different aptamers with different affinities. On the left side, for the passive reset, you can see that when we put the sensor in the protein solution, we got the signal for the bound signal here. We put the sensor into the buffer to see how passively the signal would be regenerated. For example, here for the low affinity, you can see that around 70% of the signal have been regenerated in we can see when they put the signal, the sensor into the buffer, there is no significant change into the signal. However, for the active reset, the story is totally different, because no matter what's the affinity of the receptor, when we put the sensor into the protein solution, we can perform the active reset and reset the signal to an unbound case, and then do the measurement in whatever affinity it is. All right, to finding out the mechanism behind the dissociation, we perform molecular dynamic simulation, as you see here, and we simulated the non-covalent interactions between the protein and the receptor in two different scenarios, with oscillation and without oscillations. As you see the results here, we calculated the number of non-interactions for different non-covalent bonds. For example, for no oscillation, you can see the hydrogen bonds is increasing over time, but with oscillation, the number of hydrogen bonds actually is decreasing over time, meaning that the protein and the receptor are dissociating from each other. We have also done some other evaluations. For example, we evaluated the receptor SASA, which is solvent accessible surface area, and we saw that actually for the oscillation, the SASA value is higher than no oscillation, meaning that the receptor has more access to the solution because it is detaching from the protein. We evaluated the RMSF, which shows the stability of the sensor, which shows with oscillation, the sensor is less stable. And we have also calculated the protein water and the receptor water hydrogen bonds. And we can see here that with oscillation, the number of these hydrogen bonds are higher than the no oscillation. It means that because the receptor and proteins are detaching from each other, they are making new hydrogen bonds with the solution. Generally speaking, through the molecular dynamic simulations, we found that by applying alternating potential and oscillation, we are kind of inducing dissociation with an excessive drag force and inertia force. All right. Because we are applying an alternating potential and we are oscillating the sensor, there are two important parameters that play important role for the dissociation. One is the range of voltage that we are applying, and the second is the frequency of the oscillation. Here we can see that with when the range of the potential is very low and the frequency is very low, there is no change into the signal because we are applying low energy to the system. But when we increase the level of voltage, the potential and the frequency here, we can see how the signal is decreasing from the bound case to the unbound case over time. We have done some other experiments for the low affinity, medium affinity, and high affinity for different ranges of the applied voltage and frequency, and got these phase diagrams through which we can find out the regeneration percentage of each aptamer. All right, so having the active reset methodology in hand, we perform the continuous monitoring of MPO protein in different concentration from 10 picogram per ml to 1,000 picogram per ml over 12 hours. We extended the experiments targeting different proteins such as IL-6 and TNF-alpha cytokines and measured these proteins in real-time fashion as well. We also showed the versatility of the methodology by using some other sensing approaches such as antibody-based actually sensing targeting IL-6 and also EAB sensors targeting doxorubicin, and kind of show that the active reset is versatile and can be used for other sensing approaches as well. All right. To measure the proteins and track the proteins continuously in vivo, we integrated 3 different electrodes, the working electrodes, reference electrodes, and the counter electrodes into a hollow macroneedle, 3D printed hollow macroneedle, and the macroneedle through the capillary force is able to sample ISF from the dermis layer of the skin. Here are some images for the macroneedle that have been printed using the nanoscribe 3D printer to make sure that we accurately print the features into the macroneedle. So the macroneedle is inserted into the skin of the animal and would sample ISF from the dermis layer, which actually has an excessive amount of ISF. So to make sure that the macroneedle is functioning properly, we have done some fluid dynamic simulations for the worst case scenario. By the worst case scenario, I mean when the pressure inside the dermis layer is in its minimum value, which is minus 4 millimeter of mercury. And through the fluid dynamic simulation, you can see that the main channel of the macroneedle will be filled in less than 5 milliseconds. All right, after designing the macroneedle, we fixed the macroneedle by printing some other parts as you see here, and we inserted the macro device on the dorsal skin of the rat as you see here, and we tracked the inflammatory biomarkers such as TNF-alpha and IL-6 cytokines. So before doing that, we performed some characterizations on the macro device to make sure that insertion of the macro device on the skin of the rat would not induce any inflammation in different organs, and also there is no local and temporary inflammation through the qPCR results, tracking 66 different genes. For example, here you can see that there is no difference between the control skin and the skin in which the macro device is inserted. All right, for the animal study, we targeted diabetic rats, which are known for upregulated pro-inflammatory cytokines, and we know that with fasting, because of several mechanisms that have been shown here, the level of inflammation would be decreased, so the level of the IL-6 and TNF-alpha would decrease as well. So we followed 3 different scenarios in the animal study. One is the diabetic rat with fasting without any injection. The second is diabetic rat with insulin injection at the time 20 minutes. And the third one is the diabetic rat with LPS injection at the time 220 minutes. So we measured the level of cytokines in vivo using macro device every 20 minutes. And also we sampled ISF and serum from the rat to compare the results with the macro device results. Here are some results for the animal study. For example, here for the fasting, you can see that the level of cytokines, IL-6 and TNF-alpha have been tracked, and we can see the decreased level of the IL-6 and TNF-alpha using the macro device, and the results have been compared with the offline ELISA measurement in extracted ISF and also serum. For the fasting plus insulin, we can see that because we injected insulin at the time 20 minutes, there is a temporary inflammation both for IL-6 and TNF-alpha after injection. This is because of the penetrating of the insulin needle on the back of the rat, which kind of induced the immune system. But after this temporary inflammation, they can see how fast the level of IL-6 and TNF-alpha is decreasing. For the fasting plus LPS, so LPS is known for inducing inflammation. Again, we can see the decrease of the inflammation level over time because of fasting, and then after the LPS injection, we can see the increased level of IL-6 and TNF-alpha. All right, to summarize, we have developed a technology which enables real-time continuous monitoring of protein biomarkers. We kind of overcome the limitation of a slow dissociation for the high affinity receptors, and this technology facilitates real-time measurement with high sensitivity and specificity, and it is versatile as I shown. So no matter what's the size of the molecule, what's the morphology of the biomolecule, and what's the affinity of the biomolecule, and even what's the sensing approach, we can use these active research to track the biomolecule in real time. So I hope that I've been able to convince you that using this active research methodology, we can track a wide range of biomolecules in vivo in real time. So with that, I want to thank you all for listening, and I would like to appreciate Professor Shayna Kelly for supporting me and helping me to develop the idea of this project, and also all of my lab mates to help me to perform this project. Thank you so much.
Thanks a lot, Hossein, for a very nice talk. So we have time for questions. Who would like to start? Menno? Yeah.
Hi, Hossein. Thanks a lot for your interesting presentation. Lots of new concepts and great results. You say that the reset methodology that you have developed, that is very generic. It works with EAB, it works with the pendulum, and also an antibody sensor. I didn't understand which type of sensor it is, but it's apparently 3 different sensors. That suggests that the mechanism behind it is also very generic. And for me, the most generic principle that I could think of is simply heating, that you're locally heating and that enhances the dissociation. Did you compare by simulations or experimental methodologies if it could be heating versus the drag or the inertia that you have mentioned?
So the point is that, I have not done that, but the point is that we need to do actually oscillation to dissociate this whatever biomolecule it is from the receptor. With heating, we might induce, let's say, inflammation. So the point is that for such kind of applications, let's say we're going to track inflammatory biomarkers. If we do heating, we might kind of increase the inflammation, and so we can actually.
Yeah, that's clear. You don't want to heat, but you might heat. Did you consider that your oscillating principle might cause a thermal, a thermal step?
Yeah. So I won't say that's. So it's not actually functional, but there are lots of actually steps that can be done for resetting the sensor, regenerating the sensor. Heating is one of the ways, but consider that we at the end of the day, we're going to use it into the body, right?
Yes, okay. But my question is more about what is really happening during the settings that you have chosen, the 100 hertz and the 0.2 volts. What is really happening at the molecular, at the nanoscale level? But I think we can otherwise discuss it online. I would like to give also other people a chance to ask a question.
That's actually a very interesting point. So that's why we have done the molecular dynamic simulation to see what is really happening. So through the molecular dynamic simulation, we found that so first the dissociation is happening there. The second, we calculated the local energy that we are applying to the system. So we calculated the energy on the receptor, we calculated the energy on the protein, and because at the end of the day, we are applying energy to the system, right? So, and we calculated how the receptor is receiving this energy, how the protein is receiving this energy, and because of this applied energy, these two are detaching from each other. We might be able to do so with some other actually methodologies like it's a heating that you just mentioned.
Okay, thanks, Hossein. So we have a question online.
We have a question online. So Rito is asking. Actually, they are mentioning that they are also working in a similar field, and they are having a hard time stabilizing the signal in vivo experiments. And they're wondering if you also see those things, and they are also wondering if you can suggest, share some feedback or suggestions.
Yeah, of course, when you actually talk about in vivo, so stabilizing, as just Kevin mentioned, is very difficult. So what we have done is we actually work on the surface chemistry of the sensor. So we actually use some blocking agents such as MCH, and then another layer of BSA to make sure that there is no biofouling effect and like drifting. And I should mention that we have done this for a couple of hours. So the maximum, if I'm not mistaken, 5 hours or 6 hours. So we can't do it for weeks or something because the sensor not going to be perfectly functioning for a long time.
Yeah.
Okay, so one more question from the audience here.
Hi, thanks very much for your talk. I was wondering this regeneration technique, because indeed is a regeneration method. How does impact also the binders that you have on your substrate, and how many times can be performed without losing signal in the end?
Yeah, a very interesting question. So I've done this characterization test. So the point is, depending on how much energy I'm applying to the sensor, it is different. Depending on the target, as I mentioned, depending on the affinity of the target, if I actually apply a low energy, so it can go up, it can actually go up to 40 times, 50 times regeneration over time. But if I need to apply a high energy to the system, so for sure it's going to be decreased. Let's say minimum 12 times. So for the worst case scenario, very high frequency and very high actually voltage range. So I could actually do the regeneration for 12 times and with the reduction signal of, if I'm not mistaken, around less than actually 10%.
Okay, thank you very much.
Okay, so I think in view of time, Hossein, thanks a lot for your inspiring presentation, and then we move on to the next talk, but not before thanking Hossein once more.
Yeah, thank you so much. Thanks for having me here.
And then we go to the final talk of the session by Chuanzhen Zhao. Chuanzhen, are you online? Yes, I see you. Very good. So Chuanzhen is a postdoc in the group of Sinan Bao at Stanford University. He works on developing soft and stretchable physical and chemical biosensors using organic materials. He got his PhD in chemistry from UCLA. And I see here that he was named Forbes 30 under 30 in 2023 for his work on making biosensors for mental health monitoring. So today he will speak to us about his most recent work. So Chuanzhen, could you share your screen, because then we're ready to start.
Let me share. Is it sharing?
Yeah, perfect. Yeah, it's full screen and we hear you. So please go ahead. The floor is yours.
Awesome. Thank you, Professor Peter Zijlstra. I hope I pronounced that correctly for the introduction. And thanks, Professor Prins Menno, for the invitation and the organization for such a wonderful symposium. And good afternoon, everyone from Netherlands. My name is Chuanzhen. I'm a postdoc working with Professor Jonge at Stanford. And I'm very excited and honored by this opportunity to speak today at the first continuous real-time biomolecular sensing symposium. I have been working on biosensors since the first year of my PhD, and it's really exciting to talk in this community and share some of my stories. So today I will mainly cover some of the soft biosensors that we have been developing in the Bao group at Stanford, and I will also talk about some sensor development I have been working on since my PhD at UCLA. The field of bioelectronics have provided exciting opportunity to interface with our body to monitor health conditions and treat diseases. Many groups, including ours, have devoted to develop those skin-like wearable and implantable devices to record and stimulate biosignals. Those conventional electrodes, such as those Utah array, they create severe tissue damage and immunological responses when implanted. While the softer material, they can create seamless interface with our body and high-quality signal recording without creating any significant immunological responses. So our vision is to develop those skin-inspired materials and devices that are soft, stretchable, self-healable, biodegradable, and even breathable. And here are at least some devices our group have been developing over the years. And we have developed a whole range of materials, including stretchable conducting polymers, stretching semiconducting polymers, and stretching insulating material and encapsulating materials. With those tools, we're able to build amazing devices and sensors. While most of the bioelectronic devices are still focusing on physical signals, such as electrophysiological recording and stimulation, heart rate, body temperature, and so on. However, I think most audience here today can agree with me that our body does not communicate only through those electrical signals. If you look at our body and organs, you find that the chemical signaling plays a key role in the communication throughout our body. For example, through the endocrine system, our body releases a wide range of hormones through the glands to our bloodstream and coordinate the metabolism, energy level, and even stress level. Here I highlight the HPA axis, which is our body used to regulate stress. And if you trace all the way up to the headquarters, which is our brain uses those neurotransmitters to talk between neurons. So let's take a deep dive into the brain first. So brain is, I hope the video is playing correctly. The brain is composed with around 100 billion neurons. And in between those neurons, there are over 100 trillion synapses that help communicate and transfer information. Those synapses are typically connected by those group of molecules called neurotransmitters. And when the action potential, which is the electrical signal, arrives as a presynaptic neuron, it will trigger the release of neurotransmitters. And those neurotransmitters will then trigger the excitation or inhibition of neurons in the network. Therefore, it is critical to monitor the neurotransmitters to understand the brain and neurological diseases. Here are at least two important, or maybe the most famous star neurotransmitters, dopamine and serotonin. They are so famous that I saw this short video one time on Instagram, which tries to correlate the relationships between those two neurotransmitters, or three neurotransmitters rather, with our moods. Well, unfortunately, this is wrong. But it did point out to the importance of those neurotransmitters and motivates us to develop working in this field to understand what is really going on with those neurotransmitters. My PhD advisor, Professor Andrews at UCLA, made this metaphor, and I really liked it. She said that measuring electrical pulses without also measuring chemistry is like listening to an orchestra and hearing only the rhythm, but not the notes. So let's develop some tools to look at the notes. The first tool I want to introduce today is called Neuron String. This is electrochemical sensor developed by a previous postdoc in our lab, Jinxin Li, who is now professor at Michigan State University in the United States. So this is a tissue-like soft and stretchable sensor. The sensor are fabricated with iron oxide nanoparticles and the graphene embedded in those elastomers. So the fabrication process is actually quite straightforward. You basically use a laser to generate those laser-induced graphene and polyimide, and then you delaminate them using SCBS elastomers. As you can see, we can even pattern our favorite Stanford logo and attach it on the fingertip. And with those graphene network, those electrodes can tolerate 5,000 cycles with the strain up to 50%. Our sensing is based on fast scan cyclic photometry, FSCV, which is widely used in the electrochemical community. So dopamine and serotonin, they are electroactive species, and they have a signature oxidation peak at a certain potential, which is typically around 500 millivolts. And what we did was to apply different waveforms on the surface to measure the current as our readout. So one grand challenge in this field is the selectivity between species, and those molecules have similar potential windows. We overcame this challenge by adding those iron oxide nanoparticles. With the different interaction and the charge transport between dopamine, serotonin, and those iron oxide nanoparticles, we were able to separate those two peaks from dopamine and serotonin. And as you can see, we were able to monitor the dopamine and serotonin with the sensitivity down to 10 nanomolar. So with this promising result, we then fabricated those soft and stretchable multi-channel sensors and implant them in the mouse brain, as you can see on the left, where they are one electrode with 3 channels integrated together. So here on the right, we show some optogenetics stimulated dopamine response. So those genetically modified neurons can be excited with lasers, and the typically higher frequency of stimulation leads to higher concentration of dopamine release. And in this case, we shine light on those area of VTA and implanted our device in the NAC area which is one of the main regions receiving projections from the dopamine neurons from the VTA. So as showing the data on the bottom, that we did record a higher dopamine signal with increased frequency of stimuli, successfully validated the function of our devices. Similarly, we were able to record the serotonin signals with our sensors in the mouse brain. So here on the top right, those are the signals from similar optogenetics data. And the bottom, we compare the serotonin release before and after the SSRI injection. So SSRI are selective serotonin reuptake inhibitor. That means more serotonin will stay in the synapse area instead of being uptaken. And our sensor did show an elevated serotonin level after the injection of the SSRI. Well, the first generation of the neural string has 3 channels. And our current postdoc, Mohammed, was challenging himself and tried to develop a multi-channel up to 1,000 channels fiber-like neuron strings. So he was using a simple spiral transformation to transform the thin film into fibers. And we could roll the stretchable substrate while only exposing the sensing electrode on the outside. And of course, those devices can be bent, they can be stretched, or they can be even knotted. You can see this beautiful flower that we can make from those stretchable carbon-based electrode. With this technique, our first generation of fibers have 32 channels, and we were able to get a single neural recordings in the mouse brain due to those high spatial resolution and also the soft nature of our devices. Similarly, we were able to monitor the chemical and physical signals in the GI tract of the living animal. Here you can see that we have S1 to S5, which are the recording electrode, and also SE1 to SE5, which are the stimulating electrode. The cool thing is here that you can see the spatially progressed contraction with the multi-channel fibers. You can record it in vivo. And here on the bottom, we show that SSRI injection could induce a serotonin release in the gut, and which was successfully captured by this multi-channel electrode. Now, to shift the gear a little bit, we talked a lot about this exciting part of those electrochemical sensors, and we show that the devices can be used for both brain and gut, but there are some limitations of this method. One of the limitations is the selectivity. As I mentioned earlier, that's the redox peak of this molecule are relatively close and difficult to differentiate. If you add one more molecule, then you have to separate all those peaks together. And another limitation is these are mainly limited to the electroactive species, which means you need signature redox peak to look at the signal. Now, what I didn't realize today was like this, today is actually the party for the aptamers, because all the 3 talks are about aptamer-related biosensors. So I'm not gonna talk about it as well. So my previous group at UCLA, Professor Andrews and Professor Paul Weiss, we have been developing those aptamer-based biosensors. So where we functionalize the semiconductor channel with aptamers. With a target binding, those aptamers will have conformational changes. And since those aptamers have negative charge backbone, those conformational changes will lead to charge redistribution on the semiconductor surface, which can then be amplified directly and read out by the transistor. Since this signal transduction, as you already know from Hossein and Professor Kevin Plaxco, that they are quite universal, we are now relying on the chemical signals of the molecule itself. This approach can be quite universal. And one thing I want to highlight here is the capability of our system to overcome the debalance, which is commonly known as the electrical double layer in a high ionic solution. Due to this conformational change of this aptamer within this debalance, we were able to record all the signals in high ionic strength physiological biofluids. And what we can also do is we can also tune the base pair of those aptamers to shift the response curves. And we can also, of course, change to different aptamers. Here, at least some example we have been working over the years, including dopamine, serotonin, cortisol, glucose, and also phenylalanine. So with this sensing system, we have developed some implantable neural probes for in vivo sensing in the mouse brain. So the first generation of probe were fabricated on the silicon substrate. which each device had 150 micron thick and 150 micron wide. And we have 2 transistors on the shank of those probes. We were also able to implant this device into the awake behaving mouse brain to look at the serotonin signals. In this case, we use electrical stimulation, where we put the stimulation electrode in the roughly nuclei, and we record the serotonin signal in the striatum of the mouse brain. Those nanoscale indium oxide, metal oxide semiconductor enables us to fabricate those devices also on flexible or even soft substrate. Here on the left, we fabricated those nano-ribbon arrays. And you can see in the middle, we have the real-time sensing of temperature, pH, serotonin, and dopamine, all from the single devices and with those 4 different channels. And very recently, we were able to develop a tissue-like soft probe, and we were able to monitor multimodal signal, including neurochemical and electrical signal together on a single device on living right. Now we talked a lot about the neural probes and neurotransmitter monitoring with the focus on the serotonin, which is the central component of the stress system. Now for the last piece of my talk, I would like to invite you to zoom out a little bit and look at the peripheral stress system. In the endocrine system that I mentioned at the beginning, the stress is regulated by the stress biomarker cortisol. So this opens the opportunity for us to provide real-time health monitoring in the peripheral body fluids. Quantifying the mental health is critical in understanding our mental disorders. So most current mental health diagnoses are still based on questionnaire, which are quite subjective and are difficult to quantify. So developing a cortisol sensor will provide a strong tool to understand mental disorders and provide actionable feedback and early diagnosis. In collaboration with Professor Sam Mamajad and Dr. Bo Wang at UCLA Electrical Engineering Department, we have developed those aptamer-based cortisol watch for sweat cortisol sensing. Here, just quickly go through some sensing results. We were able to detect those cortisol down to picomolar with high selectivity against some hormones with similar chemical structures such as progesterone, testosterone, and also serotonin. And also want to highlight here that we were able to develop an integrated system with flexible sensors, microfluidics for the sweat capturing, and wireless electronics, and even the LCD screen to look at the cortisol level in real time. Here we are showing that we were able to monitor the cortisol level from the circadian rhythm, and where the cortisol is high, we wake up in the morning. My cortisol level probably is quite high for California morning now, and you guys probably have a lower cortisol level before as the day went down, and we were able to record all those signals with our cortisol watch. Now, to leverage the advantage of the aptamer system, I have been building this fully soft and intrinsic stretchable sensors in the Ball Lab with the organic field effect transistors. We have also developed those hybrid integrated system for wireless communication and data acquisition. So our vision is to, in general, develop those skin-like wearable patch for multimodal mental health monitoring, and those stretchable tag can integrate multiple chemical signals and physical monitoring components together. And also we are including the on-chip computation to have the on-chip differentiation circuits and also wireless powering and communication. We're quite excited in the Ball Lab with this direction going. Now with that, I would like to thank my postdoc advisor, Professor Jonge Bao. This is a group photo that was taken maybe 2 months ago next to Jonge's house, and we're certainly a big group. I also would like to thank my PhD advisors, Professor Andrews and Professor Paul Weiss at UCLA. I also would like to thank the funding agencies, especially the NIH, BIB, F32 postdoc fellow. And with that, I'm happy to take any questions you may have. Thank you.
All right, thank you very much for a great talk. Now it's time for some questions. Who would like to start? I see a question there. Yep.
Thank you very much for your presentation. I have one question regarding the electrode that is on the skin-like material. So it's supposed to be deformable when flexible. Are there any chances that it can be ruptured if it's stretchy, like if we move the fingers around, for example? Thank you.
That's a good question. Yeah. So normally we have a substrate level layer, and also in the between have the electrode, and on the top we also put an encapsulation with those elastomers, so that you will not touch those electrode, and also we want to avoid any shorting they may have, when you wear them or implant them in the tissue.
Oh, and also, how would you connect from the skin-like material, like from the electrode from the skin into the body, or you just measure sweat, like?
Are you asking about the interconnects between the
Yes.
device and electronics? Yeah. So the interconnects has always been challenging for us, because we are always trying to develop those soft and stretchable electrode, while the electronics part, which are still based on PCB or even flexible PCB, they still have a modular mismatch between them. So one of the approach we have been working with Professor Xiaodong at Singapore, that we develop those plug and play like a Lego-like system for the interconnection. So basically, in between of them, you have this SCBS layer, and you have a gold as interconnects, and another SCBS layer on the top to make the connection. So this is this interconnects is even fully stretchable, but that's definitely still, I would say, a grand challenge in this field, how you really bridge those two parts together. And one of the vision I mentioned at the end is that we try to develop a fully stretchable system, so that we want to also get rid of those silicon-based components. So we're trying to build up those wireless circuits with wireless power and communication with the wireless antenna, that we're trying to build them with eGain. So basically you can get rid of any interconnection and fabricate all the chips on the stretchable substrate. So that way the system will be way more robust.
Thank you.
Thank you.
Thank you. Any more questions? Should we go online, Livio, for a question?
Yes, so Professor Kevin Plaxco has a question. He mentions that the slopes of the binding curves, specifically the concentration versus signal change for the FET sensors, are notably small. Given that, what precision can you achieve in measuring concentrations?
That's definitely a great question. I was looking at Professor Kevin Plaxco's all the talks with achieve the precision that you will be able to get with those EAB sensor. That's definitely something we're still looking at. So one of the things we can achieve for now for us is to tell basically low from high. So for the cortisol level, we were able to look at the low level from at night and high level in the morning. And also similar situation for the serotonin sensing. We look at the simulation, and then you can see kind of a jump in the brain. But we're definitely looking at how we can really quantify them, especially they are kind of low at the linear range, because all the signal I've been showing are in the log scale. And we are also looking at how we can fine tune the base pair to see if it can expand the linear range of the signal and can be more repeatable. And one thing to note is those sensors relatively have a high binding affinity, because we are really looking at nanomolar concentration instead of micromolar. So the reversibility has always been an issue for our system. So we have always working very hard to try to get rid of that issue. Then you can really look at reversible response at different concentrations to provide a very accurate readout. Yeah.
More questions online, Livio? Okay, I have. There are two questions in the room. I have a question as well, and I'll walk to you guys in the meantime. So I was wondering, the skin sensors, how sensitive are they to temperature fluctuations? Because you wear them on your skin, and I guess your skin temperature fluctuates, and also the outside temperature fluctuates. So, can you say something about this?
They do. Yeah. So we have a built-in temperature sensor as well. so that you can basically compensate what is going on. As I showed earlier, we can have a multiple channel of the devices. So one of them can be temperature, and also in our electronics, we look at the temperature variations, both from the outside environment and our body, because all the transistors, if you change the temperature, the mobility changes as well, and the performance also changes. That's definitely something we've been considering.
Yeah, and those are two very different types of sensors, the temperature correction sensor and your actual sensor. How do you make sure that they respond in the same way with the same time constants and the same amplitude? Is there a trick, or does it work out of the box?
So we run some calibration kind of before, like those temperature sensors, they are based on resistance. So in this case, we make some carbon materials, just run kind of a line, and then connect them together. And we calibrate the sensor at different temperature, and we basically use that as our reference.
Okay, thanks, clear. I saw multiple hands here. Could they be raised? Yeah.
So for now, you showed that your sensors are 2D. Do you think there's a possibility that this kind of dual chemical and electrical sensing can have the possibility to generalize to 3D measuring structures, for instance, that could be applicable to peripheral nerve measurements?
Absolutely, yeah. So one of the direction I saw with the spiral neural string, we kind of roll down from a 2D film to kind of a fiber-like thing. So the electrode can be kind of anywhere on the outside of the electrode. That's definitely a great direction. And also for the nerves, we have some work previously to measure the blood pressure or some nerve stimulation. They can definitely wrap around the nerve or even blood vessel to look at the signals in a three-dimensional.
So actually, initially, I was thinking something like a regenerative peripheral nerve cuff that you could kind of use your flexible electronics in the regenerative part to kind of encapsulate the nerve while it's regrowing. Is that something that you think is doable?
Yeah, absolutely. That's a great idea.
I saw more hands here. Yeah.
Do you know anything about commercial application of serotonin sensor for depression? Because it's quite a unique way of diagnosing a mental disease. Do you know anything else about commercial applications of biomarker-based diagnosing of mental diseases?
Yeah, so the main kind of interest for the commercialization for mental health monitoring is more about the cortisol. I think this now a huge market that ever since we also published this paper, there are also a lot of investors are interested to look at, like, are you guys trying to develop startups? I believe my previous advisor, Professor Andrews, she was also looking at opportunities to commercialize those devices, try to look at the cortisol level in sweat. So serotonin mainly based for fundamental studies. But cortisol definitely, if you can monitor the cortisol level in sweat or in the interstitial fluids, which is, I think, more interesting recently because the issues people are having with the sweat monitoring for the accuracy. But for sure, the ISF-based sensing for the mental health definitely will be a huge market.
Okay, thank you. I thought I saw another hand here before, but.
Thank you. Super interesting talk. My question is whether the sensor and the aptamers are in actual contact with the sweat, or maybe did I miss that?
Yeah, we have a microfluidics, and when generate sweat, it'll go through the microfluidics and then with the device. Yeah.
Okay, thank you.
All right, so leave you more questions online? No? Okay, so last chance. Anybody? No? If not, then I propose that we go here locally to the exhibition and to dinner, but not before we thank the three speakers again for their great contributions. So thank you very much. Yeah, we're going for dinner, so we can send some to you, but I'm afraid it may be too late. Huh?
Good evening here, good afternoon, I think somewhere else, following online. Welcome back to this second session of this symposium. My name is Maarten Merkx. I'm a professor of protein engineering here at the Eindhoven University of Technology, also working on biosensing. I hope the people here in the audience enjoyed their nice food and also the interesting poster session. But we still have 4, I think, very interesting lectures to look forward to. And the first speaker, I think, is the person that needs least introduction. It's actually Menno Prins, who I think everybody knows, especially also in this audience, as the initiator of sensors, as the coordinator of consens, but also, I think, the initiator and organizer of this first meeting on continuous sensing. Just a very short introduction. Maybe Menno, I think, is a physicist by training. Worked for quite some time at Philips, developing also a new biosensing technology over there. But now for some time already is affiliated with our university, both at the Department of Physics, but also the Department of Biomedical Engineering, where he's really pioneered the use of particle-based detection methods for continuous biosensing, among others, and also has initiated a company that is trying to translate this technology towards applications. Well, what I just said is actually also, I think, the title of his talk. So without further ado, I'd like to invite Menno to take the floor.
That's the microphone working now, and now the slides. Okay, thank you, Maarten, for introducing, and thank you all here for still being here and wanting to learn about continuous biosensing technologies. I'm going to speak about work that we're doing here in Eindhoven, and it's a particle-based technology with single molecule resolution, aiming on the long term for many, many applications, but the industrial ones are the ones that I'm going to speak about in this talk, and the medical applications, that's more for the future, further away. Many people have been contributing to this work, so I would like to thank them wholeheartedly. It's people from the Eindhoven University and also from Helia Biomonitoring, and funding from many different sources. I'll start off with a brief introduction. How did we get to this approach of using particles? And then what is exactly this biosensing by particle motion? And then I want to do a brief step back and what is actually real-time biosensing? When do you consider a biosensor a real-time biosensor? Then I'll give an application example and a sensor that we have been developing for that one. And I'll look at the topic of precision. And there's signal precision and concentration precision. Signal precision is lovely for a technologist, but for someone in the application, it's only concentration precision that matters. And I'll speak about that. And finally, our biosensor is based on particles. And I raised the question, can we view every individual particle as an individual sensor or not? There's many dynamics processes in the around in industry, environmental, and us as human beings. And if there's a process with variable dynamics, variable unpredictable dynamics, then it's lovely to have measurement data, to have hard data so that you can control and regulate. Now, if you presently want to do a chemical determination of a certain molecule, you go to the lab and you do, for example, an ELISA or an LCMS. And this gives lovely sensitivity and specificity, but there's not a continuous data stream, and the price per data point is high. Now, if you want to move to continuous monitoring, that is already possible, mainly with spectroscopic sensors like near-infrared and Raman. And they do give continuous data, and the price per data point is good, the cost efficiency is good, but you don't get the sensitivity and specificity that you're used to in a centralized laboratory. And the aim to target here is develop sensors that tick all the boxes, that are continuous, good cost efficiency, and also very sensitive and specific. And I think that's what the symposium is about. And then you want to aim to measure many, many substances, which have very different concentrations, and also chemically they have very different natures. Sensors are available for glucose, but not yet for the drugs, for the proteins, for the nucleic acids. They are upcoming, but they're not yet available in society. Now, we've seen the electrochemical aptamer-based switches elucidated by Kevin Plaxco, and tomorrow Tom Zhao will speak about the fluorescent molecular switches. These are two examples of switch-based sensors, and the switch is handy because if it is reversible, you can use it for a long time. So in a way, we are also developing a switch-based sensor, but it's based on particles. So at TUE, we have an approach that we combine biomolecules and chemistry with nanoparticles, with optical detection principles, and we do a lot of data analysis and modeling and simulations. Now, why do we use particles? Because particles have a strong optical signal, so they're easy to detect. Then they're easy to biofunctionalize and handle in the lab. And very importantly, they're so small that their properties change with single biomolecular interactions. A property like their color, the spectroscopic shift, or a property like their motion. And we are looking at single molecule technologies, and I'll speak in this talk about biosensing based on particle motion, but on the group there's also a lot of work on fluorescence and on plasmonics. Now, this principle of developing a sensor based on particle motion, we started on that approach I think exactly 10 years ago now. And we started publishing in 2018. Now there's a whole family of publications, and there's 3 that I want to highlight because I'll speak about it in this talk. It's the dynamic concentration profiles. I'll speak about concentration precision and also about heterogeneous single molecule sensors. So what is this biosensor by particle motion? So there's a sensing surface with many particles, hundreds to thousands of particles. And these particles are close to the surface, so they interact constantly with the surface. Now the particles are biofunctionalized, and also the surface is biofunctionalized. And now we have in solution these molecules of interest, which are sketched here in green, that can interact with both the surfaces, the surface of the particle and the surface of the substrate. Strange wording, but... and now, due to this interaction, something can change with respect to the particle. Now let's look at an animation. So I start the animation. We have all these particles, and they move because there's Brownian motion, thermal motion, random motion. And if you look closely at the interaction between the particle and the surface, that's where these molecules on the particle and the molecules on the surface, they interact with the molecules of interest in green. And in this case, it's a so-called sandwich format, sandwich sensor. So these molecules in green, the ones of interest, they can interact both with the binders on the particle in blue and with the binders on the substrate in pink. And once in a while, you can get the formation of a single molecular sandwich bond. And this changes the motion of this particle. Initially, it was held close to the surface only by this flexible DNA molecule. But now, in a way, there's a second link, a very compact link, in this case, of proteins. The antibodies are a protein, and the target is sketched as a protein. This is a compact molecular arrangement that forces the particle into a different motion. It can wiggle a bit, it cannot move around anymore. So to summarize, we have particles close to a surface, in this case tethered by a flexible piece of DNA. Particle is biofunctionalized, the substrate is biofunctionalized, and then we have the molecules of interest in solution that reversibly bind to these binder molecules. And this causes switching of states. The particle can be either in an unbound state, where it moves around a lot, or in a bound state, where it moves around much less. And we look at the kinetics. We look at in which state the particle is and how fast it switches between one and the other state. And that's called the switching activity. And how do we know the state of the particle? It's by measuring the motion of it. So we look from the top with a microscope. We track the center points of the particle, we look for a certain time, and then we see if the particle has a lot of excursions or it has been moving very little. And that defines the state of the particle. So we do video microscopy, we record lots of images, we track individual particles, we look at their center points, and then we see if the particle has been moving a lot or very little, and that determines the state. So this is an example of 4 of these particles. You see the pixels from the camera. Using the pixel data, we determine the center point, and then we accumulate center points over a certain time, for example, 1 minute. And then we see that the particle left bottom has been moving a lot. On the right, the particles have been wiggling a bit, and this 4th particle left top is completely stuck. And the interesting thing is this state of motion changes over time. So in case of the sandwich assay, if there's very little molecules of interest in solution, then the particle is mostly free, it's very mobile, and once in a while it's stuck and then it's free again. When the concentration of molecules in green is very large, then you see very frequent transitions. Of course, stochastic, these are random processes, but very frequently you see that the particle moves from the bound to the unbound state and vice versa. And from this data, we can derive, using a calibration curve, what the concentration of the molecule is in solution. So this is what the real data looks like. It's very noisy data. You see the x, y positions of a single particle. It moves a lot, and then suddenly it's stuck for some time, and then it moves a lot, and it's stuck for some time. But you see in both the channels, the x and the y, that the particle is either moving a lot or it's stuck. Then you can recognize the transitions of these states, and then we can define bound states, unbound states, and we get histograms. We can look at distributions and do statistical analyses. So these are digital signals. These are switching signals from individual particles caused by and switches are caused by the individual biomolecular interactions. And the statistical analyses, they give the detailed information that we then exploit to get sensitive sensor, specific, precise, accurate, and also robust. When we started, we had this design with a particle tethered to a surface by a piece of DNA. Later, actually 4 years later, we published another version without any DNA. And now you may wonder, how come that also works? Well, it works due to gravity. These particles, if you select the right particles, due to the gravitational force, they stay quite close to the surface. Not as close as what you get with a flexible piece of DNA, but close enough that the sensor can still work. There's enough interaction between particle and surface, and it's slow enough that you can track individual particles and their motion. So these are two different variants that we use for different purposes. Typically, the free particle motion, what we call the FBPM, we use that for screening, because chemically it's more simple to make. But if you want to have a very stable sensor where every particle stays at its position, then the top version, the tethered variant, is more comfortable. By now, we also have lots of pieces of equipment. At the poster session, you may have seen the sensor piece, the sensor at Helia. It's a compact instrument, and actually the sensing part is really, really small. So we have very compact readers because these particles are easy to visualize with a microscope. So we have a microscope objective and a camera, and we have a LED source at the bottom, and then you can see all the particles. When we started off with a simple macromolecular system, and that was the sandwich format using oligonucleotides, single-stranded DNA. So we had single-stranded DNA on the particle, single-stranded DNA on the substrate, and complementary single-stranded DNA in solution. And this is how we proved for the first time that the principle works. And the interesting aspect is that the sensitivity is expressed in picomolar. So that's the typical concentration that you can achieve with ELISAs, also sandwich-based analytical assays. So this shows the sensitivity, of course, not yet all the other aspects like reversibility, like time behavior, etc. But this was, the birth of the method when we say, hey, really it could work. But this is for a big molecule that you can bind to from two sides. Right? It's the sandwich format. Suppose you want to detect a small molecule, so small that there's only space for one binder, not for two. Then you cannot do a sandwich arrangement. And the trick you can play is then do a so-called competition format. So how does the competition format work? Well, in this case, on one side you have a binder for the molecule of interest, here sketched as an antibody. And on the other side, there's not now an antibody, but an analyte analog. So you make an analog that is an immobilized version of the molecule of interest, and you couple it to, in this case, the particle. Now this sensor, in absence of the analyte, in absence of the molecule of interest in solution, the particle will have a lot of tendency to bind to the surface because the antibody, it recognizes it's complementary to the analyte. But now, if there's a lot of analyte in solution, then these analytes, they bind to the antibodies. And then the antibodies cannot anymore bind to the analyte analogs on the particles. It's like there's a competition of the analyte and the analyte analog for binding to the antibody. So this sensor, in this design, will have a lot of switching in the top situation, when there's no analyte in solution, and much less switching at the bottom, because the probability for the particles to bind to the surface has been decreased. So the readout is somewhat different, but it's the same biosensing by particle motion principle. And we studied in the early days already creatinine. That must be of interest to you. We conjugated the creatinine. We bought commercial anti-creatinine antibodies. And then we did the sensing. And the way we do the sensing is, you expose the sensor to different concentrations. And then you measure the switching activity. And then you can plot the dose-response curve from that same data. And then you see this common inverse behavior for a competition assay, where there's lots of signal at low creatinine concentration, and there's very little signal at high creatinine concentration. Now, by now we have been studying a lot of different analytes, small molecules like creatinine, cortisol, glycoalkaloids. I'll speak a lot about glycoalkaloids later because of the application in food processing. Diclofenac we're working on, and all of those we've been studying in competition assay formats. We've been working on single-stranded DNA, and now we're also detecting double-stranded DNA using DNA and antibodies as binders. And on the protein side, there's a whole family of proteins. Many of them are inflammatory proteins, where we study them both in sandwich format and in competition format. And the concentration ranges are listed below, ppb to ppm. And for small molecules, it's nanomolar to micromolar. And for macromolecules, it's in the range of picomolar to nanomolar. So how do you develop a continuous sensor? Well, it starts always with the basic design of the sensor. That means the binder molecules, you have to study the dependence on target concentration, and then what you end up with is a molecular design and dose response curve. That's the basic work of biosensing. But of course, that's not yet continuous biosensing. For continuous biosensing, you have to look at also reversibility. So you do series of increases and decreases of target concentration, and you want to see both association as well as dissociation. And then the main output is concentration time profiles. The next step is to go even deeper in the time behavior. You look at response time to get to the time delays of the system. And also long-term operation, because you're interested to see how the sensor changes over long times. The next step is precision. You want to understand the variabilities in the sensor. And you may start off with being interested in the signal variabilities, but in the end, what you want to know is the concentration variabilities, because that's what the end user will get this information and wants to see. And the main output parameter is the concentration precision, and that's described as the CVC. And the CV is the coefficient of variation, but then it's the concentration variant of the coefficient of variation, not the CV on the signal. And after the last part is always the correlation, to correlate your results with reference methods and to get to a calibration method and also a sample pretreatment method that gives you reliable output. Now in this talk I will focus on time and on precision. And time I will focus on time delays. How do you study that and what is it? And then I'll talk about precision. So let's look at time delay. If I have a system of interest that I want to monitor continuously, then the molecules from the system of interest get into the sensor by some physical-chemical principle. Can be diffusion, can be advection. And then they have to interact with the binder molecules. So that's the biochemical reaction. Thereafter, the binding has taken place, and then that binding has to be translated into a signal. So there's a physical transduction method and signal analysis, data analysis. And the series of all those steps gives you the total time delay. And you want to understand which of these mechanisms play which role. So how do all of these contribute to the total time delay? And there's two basic methods to understand or to disentangle to some degree these contributions. One is by applying a step function in concentration, and the other one is by applying an oscillating concentration profile. If you do the step function variant, you'll see that if you apply a step function, then after some time there will be a time-dependent response in terms of the physical properties, so the molecules that are binding, and then of course with some delay related to signal generation and signal processing, you'll get the final reported signal, and that one is in red in the middle graph. The right side is another experiment that you apply a time-dependent concentration profile, a sinusoidal profile, and you look at the concentration output, and then you see to what extent that is time shifted with respect to the original applied concentration profile. But to do one step back, when is a biosensor a real-time biosensor? what kind of time delay would you feel allows the wording real time? Is it a second, a microsecond, 10 seconds, half an hour? Well, you may have an opinion, but if you look at the use of the sensor, then actually that's what tells if it's real time. Because if you're looking at glucose levels, then actually a sensor that responds in 1 minute or in 10 seconds is real time, because the fluctuations of glucose, they happen on longer time scales. But if it's about an inflammatory process that takes maybe half an hour or 1 hour to develop, then a sensor that reports data after with a time delay of 10 minutes is already real time for that process. And maybe for another process which happens on a time scale of a millisecond, you would like to have a sensor that responds on 10 microseconds. So it depends on the application. You cannot generally say, taking only the sensor, that it's a real-time sensor. It depends on how it combines with the application. So we try to formulate that in a definition, and in this paper we propose to define continuous monitoring as a process and a technology to continuously collect measurement data from a system of interest with a well-defined frequency and time delay, where the frequency is the rate at which you take the data and the time delay is the difference between the time at which the measurement result becomes available and the time at which the system of interest was really in that condition. Then the continuous biosensing is to do continuous monitoring with the biosensor. And now the real time, when is it a real-time biosensor? When are you doing real-time biosensing? Is when you do continuous biosensing with a time delay that is small with respect to the timescales of typical fluctuations in the system of interest. So now we can get to this experiment where we said, well, let's apply a sinusoidal concentration time profile. So we took two Sirius pumps, one filled with high concentration, the other one with low, and then you can control these two pumps and then cause a sinusoidally fluctuating concentration profile in the sensor. And with absorbance, we could see how the concentration would vary in the total measurement chamber, and with the sensing surface, with the particles, we could see how the concentration developed at the sensing surface. Well, this is just an example of one experiment where on the top you see the time-dependent concentrations that we applied, and at the bottom you see that if the frequency is low, that means I think the period is 10 minutes, then you see that the sensor follows quite nicely the applied concentration profile. But if you go faster, in this case it's 1 or 2 minutes, then you see that the amplitude decreases and the phase shift also increases. So it acts like a low-pass filter, and you see that for this sensor design with this chamber and these antibodies, there's the total delay time causes this frequency behavior. Okay, so this is one of the ways to study the dynamics of the sensor and the sensor response. Now, if you would like to apply a sensor in a process, in an industrial process, then it's important to know also the terminology that people use in industry. And they speak about offline sensing, at-line sensing, and online sensing. And you see on the sketch that the offline gives the least data points per unit time, the sparse red dots. At-line you get more, and with online you get even more. And this is, step by step, getting more and more data and automating it more and more, where offline, of course, is take a sample and bring it to a lab, and online is where automatically samples are taken, automatically samples are pre-treated, automatically measured, and then maybe also the process is automatically regulated. And which problem can this solve? Well, this can solve a problem of variability in the industrial bioprocessing, because food, biotech, and life science companies, they deal with variations of their biomaterials and variations of their processes. And this causes fluctuations in their quality, yield, and productivity, and then corresponding costs. And the question is, how can the resulting inefficiencies, such as loss of product and loss of resources, how can that be avoided? Well, the ideal would be to have a process that is continuously regulated, because you have continuous measurements with a short time delay and then immediate action on the process. So that you would have a regulation without a delay, or without delay on the timescales of that process. It would be automatic, data-driven, you could control the timing of the process, and this could improve then the efficiency and the sustainability. And now I have one example in this field, and that's the area of protein extraction, in this case from potato. Plant-based proteins are very important because they have lower CO2 emissions, lower energy use, lower materials used than animal-based proteins. And one of the sources of plant-based proteins is potato. With one of the challenges is that the plant makes its own so-called secondary metabolites, its own defense system. And these are bitter compounds. And it's not rare, strange that we find them bitter, because also animals will find them bitter and then they will not eat the potato. So it's their defense mechanism. But if you extract protein from potato, these bitter compounds, they stick to the protein. So you have to get rid of them, and that's a process that the industry would like to regulate. Now, these are compounds with a steroidal group and a sugar-like group. And it's the steroidal group that is common. So we raised antibodies against this common steroidal group called solanidin. We raised phase display antibodies. We put those on a particle, and on the sensor surface, we applied the solanidin analog. And using DBCO chemistry and peel pack surface, it's all described in this analytical chemistry paper. And last week, this paper came out where we have done a huge statistical study on the performance of these sensors. So we have a sensor, and this is very similar to the instrument that you may have seen at the poster session with the HELIA biomonitoring. There's a sample handling unit, and there's the sensing unit. Oh, wait, you don't see the... Oh, wait, I have to get my pointer. So this is where the cartridge is. This is the imaging camera, and this is the sample handling unit. You can put in many samples, and they all flow one after the other over the sensing surface. And in the sensing surface, you have the cartridge with the particles, and these particles, they have the anti-glycoalkaloid antibody on the particle, and on the surface there's the glycoalkaloid analog. And this allows automatic measurements of many, many samples after each other. So these are 8 measurement cycles, in a way measuring 8 dose-response curves. And at the bottom you see one of those dose-response curves. It's like an S-like curve with this typical competition-like character, high signal at low concentration and low signal at high concentration. And now we got interested in the next topic in this development of sensors, which is precision, signal precision and concentration precision, with the real interest in the concentration CV, so the relative variation of your concentration output of the sensor. And then we realized that it's not easy to define that, because it depends on time. If you are intent to compare data from your sensor on short timescales, then you have fluctuations due to processes that play a role on those short timescales. If you intend to compare data over long timescales, like weeks, then there's many more potential interfering processes that can give variabilities. So precision depends on time. So we realized we had to look at this with a new perspective, and we did that by defining several imprecision parameters which relate to different timescales. The essential sensor imprecision relates to a few minute timescale, and on the other side we have, for example, the multi-day imprecision that relates to timescales of many days or even many weeks. And you have to do a study that over those different timescales to disentangle the underlying variabilities. And you see at the bottom that, oh wait, I'll get to the numbers later. Let me first show the data. So at the top, you see again the data that I showed you earlier, and in panel C, you see the so-called precision profile. It's precision on the y-axis, and it's concentration on the x-axis. And you see that the precision depends on concentration. And that's not surprising, because the dose-response curve is not a straight line. It has an S-like shape. So it has a certain area where it's steepest, and that's where you get the highest concentration precision output of your sensor. And then with we define this bottom point of the U-shape as the fundamental concentration precision of that sensor. And on the right, you see several statistics we've been measuring at different locations. We've been comparing many, many sensors. And the main output is that the concentration precision fundamentally of the sensor itself is about 4%. And if you assume that all the other contributions are independent, then the sample pre-treatment imprecision is 3.6%, and the cartridge-to-cartridge variations is 1.7%. So these are low numbers, and that's good, because the concentration precision is extremely important for the application of the sensor. And the sample pre-treatment imprecision tells, for example, that this is to be automated, because in this experiment we did all the sample handling by hand, and that gives errors. We are not perfect in pipetting and mixing and filtering. Machines can do that much better. Oh, you're laughing because you have been doing that a lot yourself, right? Good. This brings me to the final topic of my talk. How much do I have still? Oh, that's good. I'll succeed. So our sensor has many particles, hundreds to thousands of particles. And we asked the question, can we view those individual particles also as individual sensors? Now, if you look at it from a bigger perspective, standard sensors have quite a big surface area, like 100 square micrometers or a square millimeter. And there's many molecules interacting at the same time, and then there's an overall signal being measured. That's how ELISA works, that's how SPR works, that's how the redox sensors work that we've seen in session number 1. Now, in the central lab technologies, single molecule techniques are upcoming, where you get data with single molecule resolution, but then the output is always aggregated. So you take all the data together, and then there's a single answer. So you still get a dose-response curve as a function of concentration that relates to many interactions. But now our sensor gives the opportunity, if you're able to follow individual particles over long times, to consider every single particle as an individual sensor. And we asked ourselves, how different are those sensors? And what can we learn from that? So Chris, he's sitting in the audience here, he took a glycoalkaloid sensor and he analyzed them individually over very long times and for many sweeps of concentration. And this is the outcome. So at the left you see the basis. So all of these particles, they have these digital switches. They are moving or they're not moving, they move, they don't move. And we do, in this case, 80 measurements as part of 10 dose response curves. So 8 concentration measurements per constant dose response curve. And in the middle part, you see just a selection of particles. And we see huge differences. Some particles hardly respond. Many particles respond, but their responses over time, they are very different. But if you look at the particle ensemble mean, so you take all those particles together, you average it, then you get the curves in black, you see it's perfectly reproducible. But individual particles are not only very different one to the other, but also one dose response curve of one particle versus the next can look very different. And we wondered, how different is it, and what can be the origin? And if you look at for example, there's 3 ones with thick orange lines. They look quite the same, but if you average the 10 dose response curve of that individual particle, so that's the curves on the right, you see that the one at the top, it has a sigmoidal shape, and then there are 2 with a bell shape. And at the bottom again, you have the ensemble average dose response curve. So some particles, they behave very much like the ensemble average, but other ones, they look really different. And we wondered what could be the cause. And then we thought, well, maybe it has to do with multivalency. And maybe not every particle has equal number of binders. And also maybe just below the particle, at the surface, there's not the equal density of binders. Maybe there's simply stochastic fluctuations. So Chris developed a model where with Poisson statistics, he would have a Poisson distribution of molecules on these 2 surfaces. And then with a Monte Carlo simulation, he would have these molecules interact with each other and interact with the analyte in solution. And what you see then, let's go straight to panel D, you get those response curves with very different shapes. And the dark ones, the bell-shaped one, is when there's lots of binders on particle and or on surface. Because then you can get multivalent interactions. And if there's multivalent interactions, then in absence of analyte, you have hardly any switching activity because the particle is stuck. You need a lot of analyte, and at a certain moment the particle starts switching, and then again at a very high analyte concentration, the switching stops because all the antibodies, they are occupied. So in this way you can understand that you can get either a sigmoidal type of behavior or a bell-shaped behavior because there's not an equal number of binder molecules on particle and surface. Well, for us this was a very interesting insight, and we're going to further learn from it and maybe apply it also to improve the sensor. So this closes my talk. So I've been speaking about biosensing by particle motion for real-time continuous biomolecular sensing. We discussed the topic of real-time. When is a sensor a real-time sensor? I spoke about imprecision and about single particle analysis, and I illustrated this with an application in process control. Thank you very much for your attention.
Thank you very much, Menno. So, yeah, this is presentation open for questions. Who wants to start?
Hi. Thanks for the very nice talk. I actually had two questions, if that's okay. So the first one is related to the tethered versus the non-tethered particles. Do you see that the non-tethered particles also flow off the sensor, or do you use something to still confine them to the sensor?
If you apply strong flows in our flow cell design, that would be like well above 100 microliters per minute, then you see that the particles start drifting. And one solution is to reverse the flow once in a while, but still this drift becomes cumbersome. So yeah, the solution is to stay well below the 100 microliters per minute. For example, 20 microliters per minute. Well, the guys behind you know exactly know the numbers for the different cell designs. Of course, for example, the flow cell height depends a lot also.
Okay.
Plays a large role. Yeah.
And have you tried, because I understood from one of your PhD students that the core is magnetic.
Yeah.
Have you used that to try to confine the particles on the sensor?
We did not, because if you start magnetizing the particles, they start chaining up. So that's not an easy tool to manipulate particles.
Okay.
We use the magnetic force to manipulate during the biofunctionalization steps, and in the sensor we use it, because of the mass density, which keeps the particles close to the surface.
Okay,
yeah.
Thank you. And then the other question I had was on, do you observe that you can detect lower concentrations for the competition versus the sandwich?
For the doubles, we can compare it because we did for the single-stranded DNA both competition sensors and sandwich sensors. And just like with ELISAs, with the sandwich sensors, you can go to lower concentrations by several orders of magnitude.
Okay, and it's not a negative effect that you have on the top of the particle. You also have detection sites that cannot really reach the bottom if they're tethered. Or is it
that would be a problem if you have depletion in the sensor. But then, then you would have to design it for depletion. Generally, we don't design them for depletion. So the loss of some molecules because of the top of the particle is then not a hindrance.
Okay, thank you very much.
Thank you. Are there other questions? I exercise a lot too.
Yeah. He's a marathon runner, so he doesn't mind.
Thank you for the clear presentation and the interesting insights. I have a question regarding the competition assay. So you showed some assay designs where you have the receptor on the particles and then the analog on the surface, but also other way around. Do you have, or did you see already, like in which occasions you would go for one assay design compared to the other?
Well, actually, that's driven by the challenge of the conjugation. So sometimes it's you find a chemical way or physical chemical way to couple easier to a surface or to the particle. We don't see any principal difference for the mechanism. The mechanism is driven by affinities and by densities. Affinity of the molecules and density of the molecules, and not necessarily by exactly their location.
Yeah, okay, thank you.
Thank you. Are there other questions?
There's a question here.
You guys are just trying to
yeah.
tease me, I think. Where was it?
Is it online?
With tethered method, why can there be more than one connection through the target?
Can you say it again? Sorry.
With tethered method, why can there be more than one connection to the particle through the targets?
For both tethered and non-tethered, you can have more than one connection to the particle because it depends on the density of the binders on the particle and on the surface.
Then why is it processed as either connected or not? Won't there be less movement if there is 2, 3, and
Yes, there is a motion difference between 1 and 2, and also when more, because but the difference becomes so small that it becomes much more difficult to detect.
All right, thank you.
Yeah.
Thank you. Other questions?
I think the online is also a question.
One short question. Do you have any insight that how many of the particles are non-specifically bind to the surface?
That depends on the assays and the stage of assay development. I don't know it by heart, but I think the people behind you, they can give you all those numbers.
Do you have a rough number from the back of your mind?
If you start off and you do the wrong things, then maybe everything is stuck.
Yeah, okay, thank
and
you.
And at the end, for example, with the glycoalkaloid assay, maybe Chris, you can answer. Yeah, let's say 10%, 5%.
Okay.
Yeah.
Thank you.
Thank you. I'm actually looking, there's probably also some questions online.
So Wouter he mentioned that it was a great overview on the importance of the sensor characteristics to look when considering going to market. And he is wondering what's the dynamic range that can be reached with those sensors?
The dynamic range, I would say, is typically towards 2 decades. That's what you easily get with an affinity-based sensor. You might get more, but you always have this U-shaped precision profile. So it depends on the imprecision that you would accept for the application, how wide the useful concentration range is for the sensor.
That's it. Also looking at the time, I think it's good to finish this discussion here. Give Menno another round of applause.
Thank you.
And then I hope our next speaker is present online. There she is. You're still muted, I think, Heather. Or is it possible? Maybe it's at our side.
My mic is on.
Yes, now we hear you.
Okay.
Welcome. It's a pleasure to introduce you, Professor Heather Clark. She is the director of School of Biological and Health Systems and of Engineering at the Arizona State University. I think since recently also the senior associate dean of engineering at the Health Medical School. But most importantly, I think she's an associate editor of ACS Sensors. So she's a colleague of mine, which is, I think, the primary journal in this field. So that's a little bit of an advertisement for the journal. But I'm very much looking forward to your presentation, Heather. Please go ahead.
Well, I am actually having problems connecting. I'm not quite sure what to do. I can't get my slides to come up.
You do hear us, right?
I do hear you, yes. I am not getting my slides to come up. Darn. I'm so sorry. Let me disconnect and come back.
Okay. Wat was het probleem? Er was al disconnected vorige keer. Beetje paniek. I'm more worried.
I'm a little worried too.
No. Yeah, there you are
I'm so
again.
sorry. So my problem is that I don't have administrator access to my own computer.
Okay.
And so I can't share through Teams.
Also not via desktop, right?
Yes.
Okay. What I suggest we do is that we continue the program now, and then we'll get back to you a little bit later. Is that okay?
Okay, thank you. My apologies. I'll get my IT person in here and I'll have it fixed.
Okay. Well, let's see how
Okay.
fast that works at Arizona State University. Thanks. Okay. So we'll improvise and we'll continue to the third speaker of this session. I hope she's ready. There she is. The third speaker is Claudia Scarpellini. She's a PhD student in the Consens Consortium. She did her master's degrees in physical engineering and also in nanotechnology at the Politecnico di Torino in Italy, and is now working as a PhD student in Leuven in a group of Jeroen Lamartijn. I'm looking forward to your presentation. You want the mic, right?
Yes. Will it start on its own or? Okay. Okay. Hi everyone. It's really an honor for me to be here. So thanks Menno for the opportunity, and also thanks Maarten for introducing me. Today I'm going to talk about continuous biosensing enabled by nanoswitch-based fiber optic surface plasma resonance. As you know from the introduction of Menno, and also from previous presentations, at the moment when we want to check a patient's biomarker, we rely on batch measurements, and these measurements happen at certain time points. This means that all the information that is in between two time points is actually lost. On top of that, there's a delay between measurements and results, which is critical in contexts such as, for example, the intensive care unit or immune monitoring, where conditions can change suddenly and dramatically. However, with continuous biosensing, we can actually monitor the concentration of a biomarker over time, which enables doctors, for example, for a rapid intervention in ICU or in immune monitoring. But this is not only relevant for the healthcare sector. This is also relevant, for example, in bioreactor monitoring, in the food production sector, and in environmental monitoring. As you already know, for example, continuous biosensing could contribute in bioreactor monitoring to optimize in real time and adjust conditions in order to optimize the throughput. Or in the food production sector, continuous biosensing could help ensuring high quality and safety at all times. In environmental monitoring, continuous biosensing can detect changes as soon as they happen. So our strategy for continuous biosensing consists of a nanoswitch-based fiber optic surface plasma resonance sensor. And we have a nanoswitch that is functionalized on the gold-coated surface of an optical fiber. This fiber is as the fibers used for telecommunications, and basically light is injected to this fiber, and it's reflected back to a spectrometer. This optical detection technique is sensitive to changes in refractive index. And the spectrometer gives a reflectance spectrum that has a dip corresponding to the resonance wavelength of the system. Our nanoswitch has aptamers or capture probes that are functionalized on the gold-coated surface of the fiber. And we use gold nanoparticles that are tethered to the fiber surface via single-stranded DNA linkers. On the particles, we also have single-threaded DNA competitors. So basically, when the target is present, there is a competition between the single-strand DNA competitor and the target, which will alter the equilibrium of the cell and promote the redistribution of nanoparticles in space. this means that our spectrometer will be able to detect a new resonance wavelength that is corresponding to the target concentration. This assay has several advantages, such as that it is single step, it is reversible because it relies on affinity reactions, and it has integrated signal amplification because it uses gold nanoparticles. Also, it has potential to work with different target molecules and in complex matrices. However, if we want to bring it to a continuous context, we still have lots of challenges, such as that we lack understanding of how the plasmonic signal, so our FOSPR signal, is generated. Also, we don't know details about nanoswitches. And as Menno also mentioned before, for example, we don't know if, for example, in our target step, we get a lot of nanoswitches to open a little, or maybe only few nanoswitches that open a lot. Also, we would like to improve a lot the sensitivity of our sensor before bringing it to a continuous context. And at this moment, at this point, we had not make it truly continuous. Also, we would like to be able to detect various types of targets and also to have a strategy to compensate for changes that are not due to target concentration changes, but to, for example, fluctuations in temperature while we measure in the environment of interest. Therefore, in this presentation, I'm going to present 6 different projects. These are contributions from different people, so you will see in the footer of my presentation the main contributor to each of them. So, as I said, it's really important to understand how the FOSPR signal is generated and how it is influenced by several parameters. This can be, for example, nanoparticle size, shape, density, and also gold film thickness and roughness, or the distance between the two. With this model, we are able to replicate the sensitivity that we obtain experimentally, and therefore we were able also to use it to optimize nanoparticle size or film thickness in order to obtain better biosensors with limited empirical optimization. Okay, as you may know already, FOSPR signal is a bulk measurement, which means that it is an ensemble measurement, and the signal is actually given by the overlap of a multitude of responses from nanoswitches. So if we switch actually from bulk to single molecule measurements, we will have a signal from each of the nanoswitches. And this enables us to have information, spatial temporal information with statistical insight. So in order to apply this to our FOSPR sensor, we developed an innovative setup that embeds our nanoswitch-based FOSPR sensor into a cartridge that is connected to a pump for a continuous flow. Also, the fiber surface is imaged by the objective of a microscope, and basically we obtain that each of the nanoswitches will appear as a single particle in our dark field image. If we follow the intensity over time of these spots, we can get fundamental information about the thermodynamic and kinetic behavior of nanoswitches. That will help us to develop and tune the behavior of them. So, as you know, the sensitivity is a requirement for each type of biosensor. So we developed a strategy in order to enhance the sensitivity of FOSPR sensors by performing plasma etching on the glass core of our optical fibers. So with these fibers, we obtained a sensitivity enhancement of the 65% with still limited optimization. And we confirmed with AFM images that the reason why this happens is that we get our gold surface to be much rougher. We also simulated the FOSPR signal that we get from smooth and edge fibers, so that we validated our idea and we're able to use this model to also optimize etching parameters. So in the, let's say, state of the art of this presentation, I mentioned that we had not achieved continuous measurement. Actually, this is, we just obtained our first continuous measurements of single-strand DNA. And to do so, in order to switch from batch to continuous measurements, we embedded the FOSPR sensor into a microfluidic chip and into an automated analysis system. This system involves also a pressure pump that is able to inject a continuous flow of samples over the fiber. So, in addition to that, we tuned the competition between the single-stranded DNA competitor and the target, and this way we could achieve a reversible binding. Thanks to these two innovations, we were for the first time able to measure continuously single-stranded DNA with a long-term stability, because we could measure up to 20 hours. So the nanoswitch that I talked about now, it's aimed at DNA detection. But as you all know, there's much more to measure. So for this reason, we also developed nanoswitches for RNA and for ATP. For all of them, what we did was to tune again the competition between the single-stranded DNA competitor and the target. And there will be a compromise between the sensitivity and the range in which you can measure. So for all of them, we set a cutoff of 30 minutes, which means that the reversibility is achieved in this amount of time, and two measurements will be less than 30 minutes apart. But as I said, this is tunable according to the needs of the biosensor. In the case of ATP, we also measured in 2 times diluted cell culture medium, because our aim is to in the future do ATP monitoring in cell viability studies. So in order to be able to compensate for fluctuations that can happen in the environment of interest, which can be, for example, temperature changes, or in the case of SPR, for example, light intensity changes, we developed a self-referencing strategy. And in our case, this means that we dipped our FOSPR sensor into PDMS, which creates a reference zone that in reflectance spectrum is visible as a second dip. And this can in turn be used to correct the first dip for these fluctuations. So we can conclude that the nanoswitch-based FOSPR is a highly performant and general strategy for continuous biosensing. In the future, we would like to use our model to get a deeper understanding of the FOSPR signal generation, and to perform, again, more single molecule studies to deepen our understanding of how our nanoswitches work. Also, this can be used to develop and tune novel nanoswitches, and we would like also to integrate all the strategies that we developed, such as self-referencing and sensitivity enhancement, to our continuous measurements. Finally, we would like to apply our nanoswitch-based FOSPR sensor to real-life applications and more relevant applications, such as ATP monitoring in cell viability studies. My presentation is almost finished, so I would like to acknowledge the All Biosensors group for their constant help, and especially all the people involved in the development and the supervision of this work. And also would like to acknowledge the molecular plasmonics group, and especially Peter and Livio for their help in single molecule sensing, and also Philip Delport from Fox Biosystem for his insight on FOSPR technology. Okay, my presentation now is really over. Feel free to ask any questions. I would just like to tell one thing more, which is that I forgot to mention that about single molecule, I also have a poster on that, and about self-referencing, my colleague Zhao also has a poster on that. So if you are curious, you can come.
Thank you very much, Claudia. Open for questions. Who wants to start?
When you build your sensor and you couple the particle to the surface, do you know how many molecules there are in between, or do you have an idea of a range, or how would you measure this?
Yeah, so in the bulk measurements, actually we have lots of DNA, both in linkers, so the particle that the DNA that connects the particle to the surface, and also in the competitor. And that seemed to work well. In the case of, for example, single molecule studies, that's actually what we also want to investigate. At the moment, I did some experiments on quantification using fluorescent DNA, but I think it's something super relevant in order to, like this will change a lot the dynamics. Let's say in the single molecule, yeah, as you also mentioned, the linker would make the like many linkers, so a very multivalent situation will make our particle to be more constrained, and also then our intensity change will be much less, especially because we rely not on particle, let's say, appearing and disappearing, but rather moving, so changing a bit the intensity, but that will determine a lot the maximum sensitivity we can achieve.
Yeah. Okay, thanks.
Are there other questions? I had one myself. So you showed some kinetics data, right? So I think for DNA, RNA, and also ATP, kind of struck me in those data that they all seem to have more similar kinetics, especially. So I don't know what T reverse means, but yeah, that's probably the second part. So it's all the same.
Yeah.
Kinetic. Is that a coincidence, or are we really looking at the kinetics of the molecular interactions? Or?
So in this case, so these data are, so first of all, they are not continuous measurements, they are measurements still in bulk, these ones. And in this case, it's a very multivalent situation, I think. And I think that's, let's say, I'm not sure exactly about the kinetics, honestly, because it's not something that I investigated personally, if they would behave similarly also in other conditions. For sure, that's, let's say the shape of these reversibility graphs depends a lot on how we tune the competition, because we did, I will show you in another slide. Okay, so yes, so basically my colleague Annalise, she tested different, so in order to tune the competition, she tested the length of different aces and target, so the competitor and the target, in order to investigate to find a situation that was reversible. So it means that the second step has to reach the same signal as it had before. And as you see, it's really a trade-off between sensitivity and reversibility. So the ones that are more sensitive, they are usually the ones that are not reversible. So I think that alters a lot, maybe, let's say, the signal depending on, with respect to your expectation, maybe to see that. But yeah, it's for sure something interesting to note.
Yeah, I guess so. Kind of the classical dilemma I think we have in continuous monitoring. If the affinity becomes too high, the reversibility suffers. Yeah. Okay. Are there other questions? Yes. Sorry.
Is there a limitation in terms of the size of the molecules that you're targeting? Because these are relatively small molecules.
Yeah, that's a good question. So indeed, there is at the moment a limitation because what we do is a competitive assay, and also we have, let's say, a negative signal when the target is present, which means that if we, for example, want to bind something that has a high molecular weight, we could, for example, a bit counteract, this effect. Because we, in our signal generation mechanism, we rely on the evanescent field, okay, but also on coupling, which is a similar effect, but that happens on a even smaller scale. So this means that, for example, if we bind something with very high molecular weight, then the coupling effect could be, let's say, totally counteracted by the refractive index brought by the molecule, for example. So at the moment, yes, it has limitations. We haven't challenged it, let's say, until that point. Also because at the moment we rely on aptamers, and we do not have, let's say, a library of aptamers that is so, so big that we can test a lot. But yeah, for sure, it's something to investigate more.
Thank you.
Are there questions online? Are there still questions here in the audience? If not, then let's thank Claudia for her presentation. I don't know whether I should check whether Professor Clark is there.
Okay.
We see your presentation, Heather, so I think things are
That's
working.
fantastic.
Okay, then without further ado, I give you the floor.
All right, thank you so much. I appreciate everybody's patience, and a big thank you to Claudia for stepping up and filling in. And I will give everybody a word of advice. There is a 10-character requirement on asking for administrative access to your computer at ASU. So please doesn't work, but please does work. So now I have proper access to my computer. So I'm really happy to be here today. I thank you for the invitation. I'm going to talk about our work in imaging the chemistry of the body. In particular, we feel strongly that in order to visualize the chemical content of the body, we need to build probes and sensors that will help us understand the identity of these biomarkers, their location in the brain and body, and the amplitude and timing of the signal. So we need to really have this continuous approach to understanding quantity and identity of different biomarkers in the body. And so if you think about whole body imaging, whole animal imaging, or even humans, we really have a trade-off, right? So we can get a great image like is here on the left, an MRI image, which really shows us structure of something like the brain or other areas of the body, but it doesn't tell us what's going on. As you are listening to me or thinking about this presentation, really it's a lot more than structure, right? So the neurons in your brain are signaling an action potential. These action potentials are basically a chemical composition, right? So we have ion channels that open up, we have calcium signaling within the cell, we have a release of neurotransmitters from one cell to the next cell, bridges that small gap in there, and then starts an action potential on the other side of the system. And all of this is going on very, very fast, over and over again in your brain right now. But if we take just an image, all of that is lost, right? So what we really want to be able to do is create these nanoscale probes so that we can see what's going on in the right, but with imaging modalities such as the left. So our approach is to develop novel probes for biochemical detection. So we want to first identify what is the biomarker that we want to measure, and then build a probe, a nanoscale probe that will put together all of these different components that we need. So we need to be able to have a recognition unit that is going to bind to that biomarker of interest, a reporter to tell us that event happened, and some scaffold to hold it all together, because we can't just launch those two units in the body and they won't work together, right? They're going to just diffuse apart. And then in the case of the sensor I'll talk about today, we also put in a targeting moiety so that it goes to the part of the body that we need it to be. We then really take a design approach to say, okay, so we have a lot of different imaging modalities that we could use. What is going to answer the fundamental biological question that we're asking? And everything is a trade-off, right? So spatial resolution and temporal resolution are great with modalities such as fluorescence imaging, and imaging depth and being able to take a measurement that is doesn't require surgery, let's say, is fantastic for MRI, but we lose then the spatial resolution. So everything is a trade-off, and we really try to come at it as what is our fundamental question and what imaging modality do we need. We then build our probes and characterize these both on the benchtop and in animal models. And then eventually, someday, our goal is to be able to deploy these in the human body. We feel like there are a lot of benefits to this sort of continuous approach and our nanoscale probes. We can bridge animal models. These probes can work in C. elegans and mice and rats and axolotls, and hopefully someday the human body. We can get real-time trend information, so not just a single point in time, but we know when levels rise and fall of the biomarkers that we're trying to measure, which can then give us the ability to know if a therapy, let's say we applied some sort of drug to the system, we know if that therapy responded. And this is, of course, a platform for eventual multiplex detection, measuring more than one biomarker at a time. So this is a session on fluorescence. So I wanted to highlight some of our work that we used with a fluorescent sensor for measuring acetylcholine. And this was really a very interesting project for us because we were part of the National Institute of Health SPARC initiative. And SPARC stands for Stimulating Peripheral Activity to Relieve Conditions. This was a international consortium of investigators, all the way from clinicians to fundamental biology to probe makers like myself. And the question that the NIH was asking was, can we use electrodes in order to stimulate the immune system? And so basically what they were saying, stimulate organ function. And so basically what they were saying was that electrodes and microelectrodes have become extremely mature, reliable technologies, and there is a lot known about implanting these in the body. Clinicians can do this quite regularly. So since the organ systems of your body, the heart, the bladder, any of the organs, rely on nerve impulses from the brain to tell them to function. If that nerve impulse is somehow destroyed, let's say, it's not, there may be severed, something like that, right? Can we use an electrode array as a proxy and use that to stimulate the organ itself? And okay, so this is really interesting, and there's a lot of people working in this space, but why are we part of this initiative? We're part because the dose of an electrode that has been implanted on a nerve that is innervating an organ is not known. If you want to take an oral pill, we know a lot about how to figure out what is the right dose for you. But we have no idea how to set a dose for an electrode that is stimulating a nerve that is controlling an organ. So where we come in is giving a tool to clinicians so that they can measure the amount of acetylcholine that's released in real time when placing these electrodes. So basically, they can set their electrode stimulation parameters based on real-time output of what we are reading out for them. And so in particular, we're reading out acetylcholine. This is the small molecule, the neurotransmitter that is released from one nerve cell to the next as an action potential is propagated through the nervous system. It is also the small molecule that signals to stimulate the nerves in your body. It has to cross a very small gap between the cell and the organ system itself, and bridge that in order to hit receptors on the other side. So it's kind of the the space that we need to be in and why we use fluorescence, because spatial and temporal resolution are really important. So we went through the design requirements. We had to decide how big our sensor could be, what is the dynamic range that we needed it to be in, the sensitivity, and in this case, I'm defining sensitivity as being able to measure acetylcholine on a background of acetylcholine, so small changes up and down in a baseline of acetylcholine, and of course, reversibility. We then need to deliver it to the body, get it there, target a particular space, and then image through the tissue. We chose fluorescence because we're really willing to give up invasiveness in this model, because the eventual patient, not that we're at humans yet, but the eventual patient is going to have to undergo surgery anyway for implantation of the electrode. So it was okay. We could give up invasiveness in order to get our temporal and spatial response. We decided to take a page out of the literature for DNA nanotechnology as our programmable sensor scaffold. And so there's been a lot of work in designing DNA origami shapes. And there's software online. Anybody can go online and pull up something like CAD Nano and design any sort of structure, shape, design that you want to. Just a lot of fun for a Friday night for those who are interested. And so we can basically create a template and staple strands and put these into a solution together, anneal them, so warm them up and then cool them down together, and they self-build any sort of structure that you have pre-programmed with this software system. And this really is very advantageous for us because it's, first of all, every single one of these structures looks exactly like the next one, which is really hard to do with a lot of other materials such as polymers. And it also gives us a really diverse functionality so that we can build in click chemistry to the end groups of our sensors and then click on all sorts of different reporters and recognition elements and targeting structures. And most importantly, each one looks exactly like the next. So we built the nanoscale probe to measure acetylcholine. Let me see if I can pull up my pointer here. Oh, I can't. Okay, so basically we have this acetylcholine receptor that we want to target on the cell membrane of the organ or the tissue. We can build this little scaffold here. It looks like a little dendrimer. That dendrimer at the end of each of the branches has acetylcholine esterase and a reporter. That reporter is fluorescent. And it is pH sensitive. And so basically how it works is that when acetylcholine is present, acetylcholine esterase degrades it, and it creates a very localized change in pH. And this, it doesn't change the pH in kind of a bulk scale overall. It really is dependent on being very co-localized with the enzyme there at the end of each of the DNA strands. And then at the other end, we put in a targeting moiety. It's a bungarotoxin. It's a kind of shortened segment of bungarotoxin that also contains a reference dye. And so this is a fluorophore that does not change in response to pH. So it is always there as an internal standard so that we can make a quantitative measurement. And so the way that this works is that we add it to the area of interest. It self-attaches to the acetylcholine receptor, and it is there to report the acetylcholine that is released from the tissue. We fabricate this from the bottom up. So basically, we create strands, single strands of DNA, and attach our fluorescent reporter. In this case, it is FAB, which is a fluorescent pH reporter. And we have other terminal groups on the ends. We then anneal it and create the structure. And then as a second step, because we can't anneal it with the proteins on the ends because we will degrade the proteins, We then add through click chemistry the acetylcholine esterase to the structure that has been formed, and the BTX, the bungarotoxin, to the ends. And the bungarotoxin has been tagged with our internal standards. So, and because click chemistry is, so fantastic, we can just very precisely attach these different proteins to very specific positions on this DNA scaffold. As we expected, the yield is very high for every DNA scaffold. We have designed 4 attachment points for our enzyme. We get just under 3. We get just under 4 of our fluorescent reporters and just under 2 of our targeting moiety. So pretty exact from sensor to sensor. The reversibility is, of course, one of our really important points here, because we really want to be able to measure the rise and fall of acetylcholine. So if we basically attach these in a solution, and then we flow different amounts of either just buffer with no acetylcholine or acetylcholine over the sensors, you can see as it goes, the red is just buffer and the black is response to acetylcholine. It goes up over and over and over again in response. So completely reversible. And then just because, people always ask, so the example I'll show you today, and most of our examples in which we use these sensors, they're not actually in the bloodstream. They are in a much more, I'll show you. It has more saline solution in the environment. But if we were to just inject them into human blood, we estimate that our half-life of our sensors before they degraded is about 7 hours. So plenty of usable time for making measurements. So let me show you what this looks like in vivo. So basically the system I described, in which the nerves innervate the organs, is true throughout the organs of the body. We chose the submandibular ganglion because it is located on the side of the neck and is very easy for us to get to surgically, especially in a mouse, which is extraordinarily small. The arrows have moved here, but you can see that the nerve bundles have the little bumps on them. Those little bumps are the SMG. The SMG are the small bits of tissue that are stimulated by the nerves to then stimulate the saliva gland and produce saliva in the mouth. So basically the nerves come in, innervate these SMG, and then trigger them to work by releasing acetylcholine. So the way that we set up this experiment was we needed to surgically expose the SMG so that we could tag them an image. We use a standard camera with just a lens camera, fluorescent excitation source. We had to use a spatula to pull the SMG attached to the nerves up out of the body a little bit. This was both so that we could basically get into the focus area of our objective, but also so that we could decouple from the movement of the body from respiration and heart rate. And it's always, I always say that's a kind of, I always feel for my students when they do an extraordinarily heroic experiment, and it all just kind of like comes down to one slide. But really one of the big challenges for this project was that even despite the fact that we surgically exposed the SMG, there was still enough tissue attached to the outside that we couldn't just add the sensors in solution and have them attach. So Michelle, who was in my lab at the time, pulled a glass needle and brought it in at a 25-degree angle in order to get it underneath the tissue and into the SMG. You can see on the very far right, this is the fluorescent image. The needle is injected into the tissue, and you can see right there she had just injected the sensors. The sensors immediately basically localize to the receptors all around the cell, and you can see them it'll happen again. There we go. The injection happens. And everything that is not bound to receptors is basically washed away. So this happens very, very quickly. And I'll not really dwell on the animal characterization, but we had to remove the tissue from the animal in order to construct a calibration curve. We held it under a HARP and added different amounts of acetylcholine and found a lower limit of detection of 228 nanomolar. So finally, what does this look like if we use that setup that I showed you and place an electrode on the nerve in order to stimulate the nerve and release acetylcholine? So basically an endogenous release of acetylcholine in a live animal. This is a single cell from one of the SMG. We have highlighted a region of interest. The arrow just before the blue and green curves is when we stimulated the electrode that then made the nerve release acetylcholine. The upper response is 3 animals, so 3 experiments in which we use the stimulation parameters 3 volts, 10 hertz, 10 millisecond duration for a total of 100 pulses. And you can see that the acetylcholine rises rapidly and then is cleared from the tissue after stimulation. The green is our control. This is the same sensor scaffold, the same Fab reporter, and the same targeting moiety. What it is lacking is the enzyme. So it is only reading pH. We have stimulated that in 3 animals, and we see that we are not creating an imbalance in pH through these stimulation parameters. So we can be pretty certain that the blue curve is measuring acetylcholine. And then just very quickly, this is dose dependent. So if you change the stimulation parameters, you change the amplitude of the acetylcholine, and you can stimulate repeatedly over and over again. And then you can also inhibit it through drug inhibitors and see that the signal goes away. And finally, you can make this quantitative. Actually, one more slide. You can make this quantitative by using your calibration curve and going back, and we find that at that first stimulation, at this 8-volt stimulation, we released about 2.5 micromolar acetylcholine at the peak. And because this is an imaging technique, I really like to highlight one of the advantages to doing imaging in a continuous manner rather than just making a single point in time measurement. So this is an SMG, same stimulation parameters. And what you can see here, we used Aqua. This was in collaboration with Kira Piscanzer. And we used her program to really map out the acetylcholine propagation across a single SMG. What was kind of interesting to us was that I had envisioned that the acetylcholine would be released from the entire, this is an extraordinarily small, the SMG is extraordinarily small. I had envisioned that it would be released across the entire SMG at the same time. But what really happened was that it was stimulated at the top, closest to where the electrode was placed, and propagated across that tissue. And so this is an area of interest of looking at organ systems such as the heart, where this propagation property might be really interesting as we try to understand more about heart disease. So summary and next directions, we really established this protocol to create these acetylcholine sensors. We want to scale up to larger animals and even humans. We also, I have to say, are very interested in working with other groups in order to provide this technology to others that want to measure acetylcholine. But what we're working on now is, if you think about our system, it's extraordinarily modular, and we use fluorescence because of the temporal and spatial properties. But what we would really love to do and are working on now is to exchange that fluorescent reporter for one that has photoacoustic properties so that we can do deep tissue imaging without having to have the animal model exposed surgically for the entire duration of the experiment. So if you know anybody with great photoacoustic probes, send them my way. And so with that, I'd like to thank my group and you for your time and take any questions you might have.
Thank you very much, Professor Clark, for sharing these beautiful and impressive results. Are there questions here in the audience for Professor Clark? Maybe I can start. So actually you ended up with this cartoon of your probe, which is a dendritic structure, right? So you have, I think, 4 copies.
Yes.
So what is the reason to go for this dendritic structure? Is it just to enhance the signal, or?
Yeah, so it's a very interesting question. We at the time we had gone smaller so that we had just one strand and it had one reporter and one enzyme on it. And it just didn't generate enough signal for us. Really it is a trade-off between being able to come near to labeling that synaptic cleft, which is really small. It's like, tens of nanometers, with balancing the number of reporters that we have on each one. 4 worked out well for us at the time, but I think that if we wanted to deploy it in other regions, making a structure that was less degradable, something that was really much more rugged might be in our best interest. But it's always kind of a balance.
Okay, thanks a lot. Are there questions here? Menno.
Hi, Heather. Thanks a lot for your presentation. I was wondering, to do a reliable measurement, what concentration of this construct do you need at the site of interest? I mean, the acetylcholine is in the micromolar, but the sensor itself, what is the molarity of that one?
So we don't really measure the molarity. So it is really for us, I think, the number of, or the percentage of bound sensors, because again, a little bit of a trade-off is that we want them to be there in the area of interest. But if we start blocking too many receptors, then we start to distort the actual biology of the system, right? So you can imagine that you could either see no response, no acetylcholine release because there's nowhere to go, or you could see the system kind of go on overdrive because you're just pumping it out. And so where we did was we basically attached just 10% of the receptors that were available to our reporters. And that to us was the right balance of being able to have enough reporters there to make the measurement, but not so many that we felt we were distorting the biology. But that is only through doing our control studies that those were not showing a change in pH. I think we would need to actually patch clamp to really, really verify that we weren't distorting anything.
Okay, thanks a lot. So it's a delicate balance, all those aspects. Yeah.
It's very hard to work in vivo.
Thanks.
Yeah.
Thanks a lot. Other questions? Also not online? Then I would like to thank you once again, Heather, for sharing these results.
Thank you.
And that leads us to the final lecture, which will be given by Professor Marien de Jonge from Radboud University Medical Center. He got his PhD from the University of Amsterdam, then was a postdoc at the Institut Pasteur in Paris. Then I think first got a career in a company, in R&D project leader for Merkx, Sharp and Dome, and then moved on to Nijmegen, where he's now a professor infection and immunity, and I think that's also the topic that you're going to discuss, so a clinical perspective on immunomonitoring.
Thank you very much. Thank you very much, Maarten, for the introduction, and thanks also for the invitation to be here. I realize that I'm the final speaker and that you're all very tired. It was a fantastic day. very impressed by all the data I've seen today. And my talk will be quite different. So I hope you will stay awake for the next 15 to 20 minutes. So I'm heading the Laboratory of Medical Immunology, and so I will speak about the clinical perspective of continuous biosensing, but start with the beginning. And actually what we see is patients with diseases of the immune system, and we try to diagnose those patients. And it centers all around inflammation. So I'd like to briefly introduce what do we call inflammation, how is it caused, to finally come to the conclusion. So it's caused by different danger signals. So our immune system protects us against threats, from environmental threats, all sorts of threats that cause injury. They cause danger signals, and these are sensed by immune cells and other cells, but primarily by the immune cells that generate inflammatory signals. And that leads to the activation of effectors, and at the same time also to the adaptation of the system of the different organs. In the end, that leads to that will take away the important cause of the danger, and then finally, in a normal case, that will resolve and get back to an homeostatic situation. And it's important that to understand that, let's say the immune system is really, and that's why it's more difficult to study. It's not centered around a specific part of the body. There's lots of cells moving from one place to another. There's different cell production sites, the thymus and the bone marrow. There's different immune induction sites where an immune response starts. And there's many cells that sense all these danger-associated or pathogen-associated molecular patterns. We call them DAMs and PAMs. Interestingly, there's different. For sake of simplicity, we separate the immune system in an adaptive and an innate part. And there's different cells that have different functions, but there's also non-immune cells involved in the sensing of these danger signals. And they are epithelial cells, endothelial cells, fibroblasts. They remain at a certain spot, but the other cells, they move around. And especially the innate immune systems, and it's also listed here, they have different effector functions like phagocytosis, degranulation, etc. So they all play a role in the first responses to a danger or pathogen-associated signal. And then now we dive a little bit more deep in this way of sensing. So there's all sorts of cellular receptors. They are localized at the surface of the cells, but also in the cell, so in internalized receptors. And as soon as there is the pattern is the molecular pattern is recognized, then there is a whole signal cascade starting to run up till and that is based on protein-protein interactions, phosphorylations, dephosphorylation, and in the end, there is the protein translocation to the nucleus. The transcription, the phosphorylated transcription factor, is finally leading to the transcription of the most important molecules in, let's say at least in the signaling, are the cytokines, chemokines and cytokines, and they lead to and as you can see, these are important inflammation biomarkers. They have a role in signaling from cell to cell, closely, or the cell itself is activated by these cytokines, but also more on a distant level, so the endocrine actions. And apart from this is all cellular based, or based on the cell products, antibodies, but also cytokines. But there's a whole system, the plasma protease cascade. In plasma, it's called the complement proteins. And this is the most conserved part of our immune system. They are, and also, you can also find those proteins in very low animals, like snails. And so that's very intriguing, I think. We studied this system for quite a bit, because we were hoping to find interesting biomarkers that could help us to diagnose patients much better. So we targeted those different components. As you can see, it looks like a very complicated system. There are many components, but they all finally end up let's say, it activates, and finally it protects us primarily against bacterial infections, but at the same time this system was evolved over time, but originally it has an important function in clearing dead cells. So it's a kind of surveillance system that is constitutively expressed throughout to protect us against, well, first of all, to clear the dying cells, but also to protect us against particular bacterial infections. So that's a lot of components, and I want to keep it simple, but it remains complex, but at least what we can derive from what we know so far is that generally this inflammatory response protects us, is well controlled, because let's say the human body or any organism with an immune system has the balance between, at one hand, the resistance caused by injury, such as an infection, and at the other hand, prevent collateral damage. Because, well, so there's a balance between the controlled inflammation, and if it goes wrong, then you can get dysregulated inflammation. So this is all orchestrated with many instruments. As I've already mentioned, the different instruments like the cells and the cytokines and the antibodies and the complement system. And in the end, when we continue on the analogy between music and immunology, you could say, well, first of all, there's the amplitude, the on-off reaction of the immune response. And secondly, there's the frequency and the timing of the different signals that are involved, defining finally the melody, the tuning, based on stimulatory and inhibitory responses. So the topic of today is primarily acute systemic inflammation, because this is quite a severe disease caused by a dysregulated immune response. It's one of the leading causes of death. It's more than, according to the WHO, more than 6 million people die yearly because of diseases associated with these acute systemic inflammation. And there's unfortunately no effective therapy available. So these patients are treated in ICU, in the intensive care units, but there's no effective treatment available. While there's many years of work already going on, and still there's a lot of work to do, but and research going on to understand how the development of these acute systemic inflammation is going on to help develop therapy. So the cause is infection, then we call it sepsis in that case, but also immunotherapy can cause these acute systemic inflammations, then we call it it's called let's say the cytokine storms, and then we have burns, surgery, but many other injuries can cause these acute systemic inflammations. And it's actually diagnosed in a relatively straightforward way. We either see high fever, or we see actually hypothermia, heart rate increases, respiratory rate increases, and this is monitored as soon as the patient arrives at the ICU continuously. But we also measure specific immunological components, such as what we see normally in these patients is an increase in white blood cells. We call that leukocytosis, or leukopenia, and let's say a lower number of white blood cells. And then, of course, the increased cytokine concentrations are associated very often with these conditions as well. So these are the things that we measure while the patient is admitted to the ICU unit. So what is actually happening? So there's first of all, in an inflammatory response, you see an early response based on the production of these cytokines. Then a little bit later, these cellular immune responses, more long-term adaptive cellular responses occur. And then finally, we are able to measure antibodies. In the case of at the same time, the body also, for the balance, we also have an anti-inflammatory events, such as there's for a long time, when there's long time inflammation, more chronic forms of inflammation, you see also that there is certain cells that are exhausted. There are certain expressions of molecules that we can measure to determine these anti-inflammatory effects. And so in our clinical laboratory, we get samples of these patients, and what we primarily measure is actually we do a multiplex measurement of different cytokines to understand, let's say, patterns, profiles. And secondly, what we do, we look at certain expression of cells on the surface of cells. We look at especially a molecule called HLA-DR, which is an indicator of, on monocytes, is an indicator of the fact that the system is into a anti-inflammatory, in a more immune paralyzed state. And the immune paralyzed state is a very dangerous state because it can cause, the body is not able to accurately respond to infections. So hospital infections can occur, and also a viral reactivation. And there's lots of viruses that we carry, latent viral infections, like Epstein-Barr virus, maybe you've heard of that. There are multiple viruses that we carry without knowing. Those viral infections can start to become reactivated and cause severe infection. And that's only the case when you are immunosuppressed. So in summary, the dynamics of an inflammatory response has two sides. So there's the hyperinflammation and there's the immunosuppression state, which leads to the most, well, let's say the severe forms of the disease. And actually we can treat them in two ways, of course, depending on the condition of the patient. There's the immunosuppressive treatment that we can apply, but also immune stimulative treatment. Both is used, but the question is when to apply, and especially based on the current diagnostics, as I already indicated. We measure profiles of cytokines and we measure certain surface markers on cells, but there is no way that we can do that in a continuous manner. So the aim is actually, first of all, to better understand the development of these acute systemic inflammation, to allow for implementation of accurate treatment. And nowadays, very often we do single time point measurements with single biomarkers, but in the end what we need is actually multiplex dynamic profiling, because a single time point doesn't say a lot about, let's say, the dynamics of a response. So we are, apart from the work on, let's say, we are very much looking forward to all the biosensors you are developing, but at the same time we make use of the techniques that we already have in the laboratory, which is primarily flow cytometry and multiplex cytokine measurements. And based on this, we actually, first of all, we phenotype the cells that we identify in these patients. We look also to the activation state of the cells, and primarily based on the signal transduction, so the phosphorylation status of certain transcription factors. And we look at the profiling of the function, so what's the effector phenotype. And so in the end, this leads to already an improved way of dynamic immunomonitoring, as we call it, improved diagnostic process, where we integrate multiple data sets. So we include, of course, the metadata of the patient. Then we measure statically what types of cells are present or what type of cytokines are produced. And then we measure that at multiple time points in a dynamic fashion. Then we integrate the data and try to find patterns to identify, to distinguish the different patients from the different states of disease where the patient is in, to finally help the diagnosis. So this is the immunomonitoring we're currently applying without having the access to biosensors currently, which could, for at least the soluble factors, help us a lot. Still, we are doing this work in centralized laboratories, already mentioned also previously, but this needs to be done, especially in the acute phase at the bedside, and decentralized, using decentralized diagnostics, because this is essential to monitor the acute systemic inflammation. We also have a role in the Consens project, and actually we learned a lot during the past years, and I'm very thankful to all of those who helped us. And Guillermo Gouveia, who's also in the audience, is still involved, and he has worked on a part of the Consens project, especially on the development of in vitro cell models, immune cell models. First of all, to improve our understanding of the dynamics of immune responses, but also to test novel biosensors within Consens. And we focused, so just a few things about the focus in the context. We focused on interleukin-6 and about to focus on TNF-alpha. These are the typical inflammatory markers already previously mentioned and also studied by others here in the audience. And we focused on sepsis, so on infection-induced acute systemic inflammation, and then in particular on the sepsis caused by gram-negative bacteria that produce the lipopolysaccharide, or LPS. So And to our surprise, there's not much information available yet about the dynamics, let's say, in the first few hours of the cytokine responses. And that's due to the fact that in the clinic generally only limited measurements are taken with many hours in between. And so the question is indeed, what is the dynamics of the response? And that's the first question we wanted to answer. And so we used for this a very simplified model, a static model, where we collected blood cells from many different donors, and we stimulated those with LPS to mimic kind of, let's say, a septic response. And then, as you can see in the right graph, you see these are 15 donors. They respond quite differently. So there were quite some individual differences. So everything was highly standardized and performed in the same way. We used very standardized methods, and to our surprise, there was quite some differences in the IL-6 responses, both based on the maximum concentrations, but also on the dynamics. Then we also wanted to understand this is the whole cascade. So going from the LPS signaling to the IL-6 production. And there's many ways you can block, let's say, the signal to see whether you can interfere and to determine whether you can kind of mimic therapeutic interventions. This is just an example of one of the, and that's let's say one of the first to block the dimerization of the TLR4 receptor that is involved in the recognition, one of the sensing receptors that sends lipopolysaccharide. And you see clearly that when you add, this is called Rezatorvit, it's also used, clinical use. Not all the compounds that we selected are already in clinical use. Some are still experimental drugs. But interestingly, you see that there is a clear effect of the timing. So the timing of the addition of the Rezatorvit for the response. So timing of the intervention clearly impacts the response. So with these models, we try to at least, and of course these are artificial models because these are all in vitro models. There's lots of the system that is not, let's say, involved in the process of inflammation, and also not in the interference, but at least this is to get as close as possible. And it's, of course, also a simple way to repeat multiple times with different individuals to have let's say, a strong result and see whether that is reproducible and to see what the individual variation is. And there's, of course, this is all a static model, so there's accumulation of IL-6, and that's the limitation of the model. And so we are now, that's work in progress, moving from a static to a more dynamic immune cell model, where cells are moving and where by diffusion we measure the IL-6 responses upon LPS stimulation. So the take-home message is that there is a clear medical need to accurately measure the dynamics of immune responses in patients with acute systemic inflammatory diseases. And that the accurate treatment of inflammation requires a frequent or preferably continuous monitoring, and that in vitro models improve our understanding of the dynamics of immune responses and enable the assessment of continuous biosensors. And I'd like to thank our colleagues from the intensive care unit, Peter Pickers and Matthijs Cox, where we collaborate a lot on this topic, and of course my colleagues from the Laboratory of Medical Immunology, and thanks to the whole Consens consortium. Thank you very much.
Now it works. Open for questions. Who has a question? Maybe I can start. So you kind of mentioned that you're still waiting for the sensor developed in the Consens consortium, but you also showed some data, and you mentioned you use conventional technology, but what kind of technology did you use? Because you showed two graphs, one of them almost seemed continuous,
right,
and the other one had some more discrete data points. But
yeah, we kept it very simple. We used simple ELISA so many ELISAs. really, Guillermo is the absolute expert in doing ELISAs. But that's a very conventional way we measured it. In the end, I mean, the next step could be to multiplex, so to measure multiple of these cytokines. But so far we have used very conventional technology.
So, but just a lot of work
A lot
on
of
one
work,
slide.
that's all, yeah.
Other questions?
You said that still little is known about the dynamics of these inflammatory markers in patients, and is it because there's regulatory limitations on how frequently and how much blood you can draw from a patient, or is it a cost issue, or
No.
simply people are not?
It's probably a cost issue, I think, although but it's certainly not the access to blood or samples from these patients, because they're continuously, so you can relatively easily actually take blood from these type of patients. It's rather a cost issue, I think, and maybe it's not. Maybe people are not convinced that it is important, especially in the first few hours upon admission, to measure the status based on these type of cytokine responses or other inflammatory markers. So we are circling around as long as there's no method, and that's the other problem. When it's a cost issue, hopefully these type of sensors would reduce the cost significantly, and then we at the same time learn how these responses work. But yeah, so it's primarily a cost problem. Yeah.
Another question is, you started off with a lecture about the basics of the inflammatory system. Thanks a lot for that, because that's always you constantly need to refresh how complex again it is. But then it's surprising that there are sometimes very clear markers that surface, like IL-6 and TNF-alpha. So what is the dominant cell type or dominant aspect of that total complex system that you think is addressed by IL-6 or TNF-alpha?
You mean the source or the target of IL-6?
Either way, how come that those markers are surfacing as are popping up as very valuable markers for monitoring people with acute inflammation problems?
Yeah, well, there is what is known so far about the cascade of events is that CRP is an early marker. And IL-6 is an early marker. CRP is evidently also found because the amounts of CRP that are produced are easy to measure. That's not the case for IL-6. Cytokines in general, they are in the low range, they are non-abundant proteins. But there is, so the liver is playing a very important role. So CRP is produced in the liver. And then there's an order of events, and especially the monocytes play a very essential role. They are the producers of the IL-6 response. That's why we always focus in inflammation conditions on the monocytes. That's why we also measure the HLA-DR expression at the outside of the surface of the monocytes. They play a very crucial central role. That's very likely the reason why we focus on cytokines produced by these type of cells.
Yeah. Okay, thanks.
Yeah.
Thank you very much. Yeah, there's 2 questions over there.
Thanks so much. My question is a bit more practical about how the medical laboratory works. So the first question is just, do you handle all of the sample types that come in? So you can measure urine and stool, saliva, blood. Are all of these measured in the hospital and all of them sent to the central laboratory? Or is it mainly just blood in the hospital?
It depends on the type of sample. We also handle feces, for example. We also measure inflammatory markers, for example, for in patients that are suspected of having a gut inflammatory disease, intestinal inflammatory disease. But it depends. We have a central place where all the samples come in, and then they divide depending on, let's say, the treating physician asking for certain tests. That depends on, so we are all together, so the labs are all interconnected. And sometimes it depends on the type of test. Sometimes it's a microbiological test. We measure the pathogen that could have caused it. We measure let's say, all the immune components that are relevant. But indeed, it's centralized, it's received at a centralized place. And then from there, it's distributed to other sites.
Yeah, so I guess if you have there's not like a blanket series of measurements that covers all the biofluids when it comes in. It really depends on the doctor who orders the test. And of course, it makes sense that you would measure in feces for GI tract conditions, but I was curious if for a patient that you want to measure sepsis in blood, in fact, you also happen to have data on other biomarkers because there's a blanket test done across the board.
Yeah, true. But in those type of patients, we measure, let's say, apart from blood, we also measure CSF, cerebrospinal fluid. But feces and urine, urine could be, but it's not a very common sample to be measured in sepsis patients. And there are a few exceptions, like for example, we also do the transplantation immunology part, and that's also very acute, 24/7. So that's a different trajectory, so different route of entry of the samples. So that also, so there's some diseases with some exceptions. It depends on also the acuteness. But you're right that we try to combine also the metadata from the patient and the samples, and combine that in a way so that we can learn from other laboratory tests that are done at the same time at another place within the same hospital, within our hospital.
Yeah.
So that's more and more, especially now with all the data-driven way of looking at patients.
Digital twins and bigger models and clear.
Yeah.
Okay, thank you.
So I just wanted to ask about timelines, and from your graphs that you showed, some biomarkers they change faster, but some really take few hours even to basically increase or decrease. So I was just wondering if you could maybe pinpoint some biomarkers that change faster, so that, for example, if I would have to develop a sensor for those, what would be the time that I could aim? That, for example, 3 hours is the right time for the response, or it has to be half an hour or 5 minutes?
Yeah, very good question. And indeed, that also refers back to what was clearly explained by Menno about what is real time. That was your question. So I think in this case, we could say, well, that's unfortunately, there's not a lot of literature on this. We were, to our surprise also, especially in these very first responses. But it is rather hours. That's actually our conclusion based on these results so far. And preferably, you would like to measure at least every hour, or every hour would be probably sufficient. And completely different from the glucose levels that fluctuate every minute. I think rather for these type of inflammatory markers, it's not the same dynamics. There's smaller differences from hour to hour, but I think still measuring every hour, so it's not continuous, but at least every hour. Or is it still continuous? Can we call that continuous? Continuous, okay. Then if we strictly measure every hour, we call it continuous. But that will be probably give us a lot of more insight, and certainly we will see changes. That's what we see at least in vitro or ex vivo type of studies.
Okay, there's still an online question.
So Walter is wondering what multiplexing level would be ideal to monitor the immune status of ICU patients, considering continuous monitoring.
Yeah, well, at least there are a couple. It's difficult to give a specific number, but there is a number of, like, let's say IL-6 and TNF-alpha, the well-known inflammatory markers that at least if we could combine, let's say, 3 or 4 of these markers in time, then we will have a lot of more insight already in the status of a patient. Yeah. And especially, and the question is, which one would you select? Well, those that gives us the most information, that shows the most dynamics. For example, people also discuss this in the context of CRP, but it's not very useful, because CRP, we know, that's what we know from CRP, it increases and it remains so the half-life of CRP is taking a while. So also recovery of the patient, you won't see that based on CRP, because it remains quite some time at a very high level. So that's actually the information you need of the biomarkers. We know as soon as we know, let's say, have more information on the fluctuations and the dynamics, then we can say, well, at least these 3 or 4 markers would be very helpful to give us some more insight into the status of the patient. Okay.
I think with this, I think your response time is becoming longer and longer. So I think we should stop here, but not before thanking Marien for his presentation. I would like to thank you all for your attention, staying quite late here in this time zone. For the people watching online, wherever you are, maybe you can have lunch by now. Maybe we can have a drink, maybe. We'll see. I don't know, Menno, do you still want to say something?
Well, just something very small. We found one of those lock cards for the social hub, so someone lost it. Is it yours? Okay, we found it. Otherwise, I think we can close. I would like to thank Maarten, and sleep well, enjoy the night, and see you tomorrow.